seems like she's holding.
Huh?
She seems like she's holding. He's holding.
Then I kind of need to see the arm. Okay.
Can you see the nozzle?
Kind of.
You need more zoom. I'm gonna zoom in on the nozzle for a minute so you can see it from my side.
This is not exactly the best way to have done what we did, just so you know.
I know, actually, that's a beautiful photo. Okay, put her name on it. So we would like a bunch more. So we need 5 for you. So, question is, Peter, do
Yes.
you guys want your course here? This will be
Yes.
next to,
Liz?
That's what the manager said. I'm waiting on confirmation from Aaron, but I believe that is the case.
okay. How many cores can we fit in this patch? We need on the order of 5, 3, about 10.
Probably not 10.
Okay, well, let's keep putting them in till we'll see.
You ready?
No. Okay, then.
Easy off.
Are these all the hardest ones to get to or the easiest ones to get
Hardest
to?
ones.
The other ones are gonna
'Cause-
make us move the porch.
Because, oh, I
And,
see,
that's gonna smush whatever's in front of us.
I see.
Do you have concerns, Lisa?
Oh. We gotta fill so many cores, it doesn't, anyway, it doesn't matter, but.
mean, at some point I'm gonna have to open the porch anyway, cause this is the only floor out on the rail.
There aren't 4 cores for Marlowe
Yeah,
on
you
this
ready?
diet.
Yep.
Sorry.
Alright.
What do you want to do, Lisa? You got 4 so far in there.
We need about 10 here, so just keep punching them.
We're gonna have to
and,
watch out.
Porch out. You got room, so do it.
They don't want them on the bubble, so they'll have, but I think it's okay to use the space and out here. I mean, the Marlowe cores, they all want them together, but they're only going to be able to get 3, so those can be those 3. So,
Okay, we're watching out.
Peter, can you explain to them that you already got 3 background cores and 3, well, we can do the math. There might actually be able to take another one, because we have some extra cores on the,
Okay.
but we also have a bad core that nobody can use, so, we can try for 4.
Yeah, I mean, again.
But
it requires
Yoho needs 5,
Yeah,
and he wants 1 for Gallardo, so that's already 6 more we've got to put in here.
I hear where you come
Yeah,
from.
yeah. We'll see what we can get.
Oh, that's a bad one.
That's a bad one also.
Now one is not available.
It's a shame.
Can we collect
They
from
didn't put
the?
together properly. God dang it, man. The porch is not out fucking.
Okay. Cooking porch, let's move.
Well, hang on, because now I'm stuck here.
Maybe if you open your jaw slightly, the porch out. Pop it out.
Yeah, from now on, porch all the way out.
Not in the bubbles, for sure, okay? Because those won't survive anywhere too close to the bubbles, I think. Will just bubble up.
Okay, ready?
Yep.
Is that on a clam?
That's the picture. Easy.
Yeah.
little pork chop would be more.
Yeah. That's it right there, that's all you can go.
Lisa, if we could please get Ford here, that would be very helpful.
Okay, can you
But then we
figure out-
would be done coring the rest of the time.
You realize you have 3 background cores and 3.
There are 8 for us in the dive plan, and so if we collect 7, I feel like that is fair.
2 cores failed.
So 1 can come out of our budget for sure.
Okay, well, can you go talk to Eulogio and find out if it's okay if I take his cores in another place?
Okay.
That would be helpful also, yeah.
I don't want to watch
Um,
it.
Why?
I can put you in.
Yeah.
So there are 4 cores in the bottom there, but one of those is patches. So,
Okay, ready?
Yeah.
I think that's gonna have too many bubbles. You're way too close to the bubbles.
Release. I'm off.
He says in the area is fine.
Yeah, then you're gonna push out.
Slightly over is fine.
Okay, well, there's 5 cores in the, let's just stop for a second. I'm gonna talk to these guys. So it's 5 cores in the bottom now. 1 for Pachi and 4 for you guys. Do you want to have me collect these now?
Isn't
And-
Pachi's already in 17?
Pachi's in 17? Oh. Okay. Well, let's collect them and see how they are and if any fall out. I'm a little worried about how gas charged the sediments are.
Yeah,
and
agreed.
So let's just try to recover these and then,
Okay. Pork chop. Pork
Watching
chop.
out for bubbles again.
Stay there.
That's us. That's us.
Yeah, that'll be on sit
you guys keep track of what's happening with each one so you'll know what you want to use them for.
Absolutely.
Let's see a picture. Might be able to work out a little more than the down. Make it easier, okay?
Yeah.
Sit. Nice picture.
Nice. Is that 9? What was the number on that?
11.
11. It is 11, okay.
11.
One, two, three, 4.
Yeah.
Wait, that's 11, so what was the number on the other one? I
The
guess-
last one was 19.
Okay.
They porch in now, the rest of theirs gonna go on the rail. Maybe 12. Can you maybe zoom in there?
A little bit more on this.
Outside, that's right.
Zoom out this.
Black ones are terrible.
There you go.
I feel like I missed one. I'm amazed the sediment hasn't bubbled up. Ooh, is that gas hydrate down there? Can you guys tell me, was number 8 in background sediment? Push number 8.
Yes, I have that as a background.
Okay, thank you.
Should be the front row of the porch.
Yeah. Is there still one tube out there?
Yes.
So these bubbles that you see coming out, we're seeing on the M3 and also visually 25 meters above the bottom while the scientists were mapping with the Sebastian this morning. So they rise in a group. It looked like a gusher even up way up above the seep. So there's a lot of methane in the water here, no question. Unfortunately, we don't have the SAGE sensor on the ROV because of its depth rate and pressure rating for some components, I think. Great. Okay, so you guys, you got your 4, and there is one more, which may go to Gallardo or Jorge.
So Patrick's got 17, and then we've got 20, 18, 12, 19.
Wait, tell me which one don't you want of the 5 that were just collected?
I was a little bit worried about the methane class rate on the last one. I think that was 12.
18 was the last
18
one.
was the last one. Or I'm sorry, the one prior to the last one. So that could have some pretty significant expansion as we rise.
Okay, so you don't want 12.
I would lean away from that one. So that would leave us with 19, 11, 20, and 18.
So we would like to, thank you, sediment scoop, this tube, all these tubes here and here, and what's left of the surface sediment.
Can you reach that?
We might have to move.
Yeah, I don't know, you'll probably need to scoot forward to do it.
I'm gonna watch out.
Yep.
So those cores were great, Doug, thank you.
Yeah, those
Really,
were awesome.
really looking good.
One or two?
I'll try 2, that way it's out of the way.
Let me watch out some.
Yeah, go for it. Just got a sturdy handle. Close the lid.
What you know?
For sure.
So if you look, there's like a big bunch of tubes. Can we zoom in down there? Back here in the surface, there were like this cluster of tubes right here. Let's zoom in. It's kind of like bacterial mat. If you can try to just take the surfaces where we haven't, I guess there's bubbles there, but through here and anywhere we haven't dug deep.
Is that you
Yeah.
target the worms itself? Right?
This, yeah, we're after the worms and all the things living in the surface sediment. There might be some caprellids, there might be dorvalid, polychaetes.
It's a picture.
Yeah, that's excellent. You guys get a frame grab of that beautiful
Sure did.
scoop? Okay, and then come up. We don't need to scoop the bubbles because if we can avoid it. Fewer bubbles in there, the better. Nice.
Is that sufficient?
Maybe one more pass over the surface. If we
Out
can.
around the same area where it's gray.
Yeah, that would be fine. Gray and worm tubes. Some combination of that. It's good. Like there's more tubes still here. Gray could be bacteria. So the amphiridids were apparently in New Zealand building tubes that help conduct the methane up into the water column. And they also, that's a lot of worms. Yay, we love worms. That should be great. You can get them in. Excellent.
Happy?
Yes.
All right.
Okay.
One bag of worms.
Where do you think you'd like to put it?
Two, you know, put it in the middle, it's easiest.
Sure.
Okay. Watching out.
All right.
I'm sorry.
Two.
Two?
Either A or B.
Okay. And that was sediment scoop number 2?
Correct. Camera in.
Yep.
Great. Okay, we would like to scoot up ahead a little and see if we can take 5 push cores in this area, in a place nearby here that hasn't been disturbed and that doesn't have clams. I mean, it can be near clams. So Eulogio's push cores. I think we have an extra core now for the Gallardo Jorge. Davide, we need one.
Yeah, that you made that.
Sorry. So what am I doing? Can I side class?
Not in the bubbles, just, it can be a little further out. We couldn't fit them right in there. And then we didn't go back for any animals, aggregations or anything like that. So we should try
For the smells and the acrobatics.
the civilians and the aggregations and anything else you see. Because that fish is pretty interesting, hasn't moved a bit. I don't think we'll catch it, but figure out what it is.
Do we have a hard target anymore?
If you want another target, yes, they have. I'll show you. There's a waypoint for this mound, and there
Right out here, correct?
should be a new waypoint just
Right.
added for up there also.
That marker where that little mound is, looking at that, there's this year.
So there's two Number 10 is red, just right here.
Yeah, I need to.
And another one of these, well, we saw, we took a seal over a bunch of mud.
She almost put it. He doesn't care we're here at all.
I see a good catch that.
Is that right, Matt? Old fish whisperer.
right. Lisa's off to do some work. The afternoon team is on. Me and Damo from Norwood University in Norway. And I've got Fernanda and Peter here logging for us. And Ben and Doug doing all the hard work. That is quite a plume of bubbles. We'd like to move a little bit, Ben.
You have an idea where you want to go?
We're going to go on core again.
That area?
Yeah, somewhere around here where you can settle.
So, just let you know I'm pushing clams.
Your what?
I have to sit on the clams just to let you know. I'm sitting at clams.
Yeah. Can we sit maybe where we
Minimize.
want a core in no clams, but maybe try and sit not on
Less.
clams. That'd be
Less
nice.
clams.
Yes, less clams.
Okay.
We're going to do some more coring. The bubbles are very bad for cores, and methane. We're at 2,700 meters, so that's 270 atmospheres of pressure.
Yeah, I'm gonna.
So those little bubbles you can see there,
Oh,
if we bring them up,
fish on this
Oh,
side.
okay.
would expand incredibly and cause all sorts of trouble in the sediment cores. So we want the influence of the methane, but we really don't want much methane in the cores.
Specimen behind you.
He wants me to land less.
Yeah.
Yeah.
Roger, all you got an updated waypoint?
If we land somewhere here, Ben, would be great, and we could
Update, thank you.
Thanks,
call
Eric.
somewhere there. There's a few clams there, though. Well, we'll core in between the clams. Oh, it's a dromalia. It's a siphonophore. Or a no, that is a ctenophore.
You said that's a ctenophore, right?
Yeah, that's a ctenophore. Oh, that's like several ctenophores.
Yeah.
Sorry, what's the name of this?
Comb jelly, ctenophore.
Oh, nice.
The tentacles.
Ctenophore with a C.
I forget if you contains CT.
Normally we see them swimming with a pair of tentacles. The number of tentacles I'm seeing makes me a little confused, so I'm thinking it's a cluster. And we often see these kind of ctenophores attached to the seafloor and then putting their tentacles out to catch food. We might end up sampling that later. We should see that, yeah, one of the tentacles is probably down attaching to the seafloor. Okay, Doug, there was a xenotobella right in front of us, where the clams are, back towards where we were. And because of Ben's fantastic landing, it's not been disturbed. So it's this creature here, if we could get a nice close-up. Thank you. Yep. Purple socks.
Purple socks.
That was our old nickname for them for many years, was finding purple socks. Yeah. Okay, we sampled a few of those earlier today. All right, we'll do a little more quick push coring here.
Yeah, I do those.
Alright.
Yeah, so we have 8 left. And I think we have requests for at least 7. Can we check with Davide and Jorge? The clams you can see everywhere are large, obviously, with their big shells, and they have quite a lot of meat inside and very big gills. The gills are full of bacteria,
There you go, thank you.
and these clams are living not by filter feeding but by using the hydrogen sulfide coming up through the sediment and farming bacteria that can break down the hydrogen sulfide for their food. The
Back. Body there, here. Back, here, it's here.
Okay.
And up there.
Yeah, it's okay. If there is time and when, yeah.
Yeah. Okay. 2.
One for
Okay, so that's all of them.
8.
yeah.
Okay,
8.
we do 8.
Watching out, watching out. It's gonna be quite way out here. Watching over here now. I'm gonna have to put you on the.
Yeah.
Happy here? Greg?
Yeah, thanks, Ben. Let's see how it goes. I mean, the risk is a shell, but the reason I think it's a ctenophore, that thing, is the way the tentacles are branching looks different to a siphonophore. But we'll maybe check it out after we finish the coring. And as to the edibility of these clams, the gills accumulate a lot of... They have a big foot, and then the gills, though, are full of bacteria. And I don't think they'd be very appetizing.
Watching out.
Okay, they look pretty good. If we can, we could put out 8 here, Doug, if there's enough room. was Halloween last night, and we had a lot of tube worms and clams in the lab that needed to be dissected, and I can tell you that they have a lot of blood. Okay, it looks like these will be pretty good cores. The Xenotabella feeding mystery is that we haven't seen one where we've been able to see the mouth, and hopefully we'll have a little more of a look and find one in a posture where its mouth is up. Normally they have the mouth down, but it's just, there's no jaws, there's no gut. They just open up into a cavity. They're very simple animals. They have a body wall, which just has a skin on the outside, then a layer of muscles, then gonad, then more muscles, and then the gut. They don't have a kidney system. They have a nervous system. We're not really sure how the sperm and eggs get out. They're males and females, we know that. When we sequence the Xenotabella, though, we get often clam DNA will come out as being sequenced as well. So we actually have to sequence the Xenotabella carefully in a different way than is often done. So some studies on the only other species known before the discovery of the deep sea Xenotabellas was from Sweden, and they figured out that that Xenotabella there had the DNA of multiple bivalves, all different kinds of clams and relatives in its body wall, not in its gut. When you open them up, you don't see anything inside. And when we first discovered these deep sea Xenotabellas, we found they had DNA of the clams where they were living. We watched quite a lot and never saw them actually feeding in any way. Plus they have this soft, tiny little mouth, so it was kind of difficult to imagine how they could actually prey on a clam, which can easily close it up. So my hypothesis is that they eat the mucus. The clams shed a lot of mucus, and that eating mucus is not a bad energy source, and that may be what sustains the Xenotabella, but we have not proved that. But the ones we collected today, we'll check the DNA of the clam against the DNA we find inside the Xenotabella. Yeah, we did take pictures in the lab, but we don't share them. We share the nice pictures. Every animal gets photographed individually, and we take tissue samples. The animals are then preserved, and they're going to end up in the National Museum in Santiago. The creatures we sampled yesterday would never have been sampled before. It was the deepest seep that had been studied off Chile, and today we broke that record now at 2,700 meters, and we're seeing even more new and different creatures today. The clam may be the same as what we found at 2,200 meters, but we'll check that.
Greg, you broke my record.
What record?
The deepest seep in Chile. That was up Antofagasta in May, I believe.
Oh, you did that?
With Aswabusas and Lauren Saylor.
Okay. Did that have clams?
It did, yeah.
Yeah. Oh. How deep was that?
I can't remember, I think maybe 2,500.
2,500. So yesterday wasn't the record?
Yeah.
Okay. Did they sample the animals?
I remember we cored biomat.
Yeah.
There's a lot, yeah, there's a lot of bacterial mat. Yeah, they must have brought up some clams.
Yeah. Oh, I'm just wondering where they ended up.
It's in the press releases, yeah.
Okay, so this is the deepest Chilean seep.
Yeah, I don't know. Yeah, that's, I don't know if the download or not that one.
Shows that you have for the rest of the line. There's this spot here that looks extremely interesting. It's like 10 minutes.
Yeah, I know, it's right on
straight
the
shot
edge of that.
from here.
I'll just open out and get this down a little
I
bit.
think it could be worth to go there,
Mhm.
and we can switch on the mapping while we do the rest.
Ah, it wasn't.
finishing.
It wasn't mapped.
It's mapped, see, kind of half of it.
Mhm.
But the mound does this and can come up here, just out. So we don't take the mapping time as mapping time, but it would be great to see what's there. I'm expecting to see some probably cost so much higher that maybe there could be something showing up either hydrate or things, because it is not the best angle, but there are big scarps and big things, so I think it could be worth going. It's not super far.
Yeah, we'll definitely head
Oh, it's-
to this,
Yeah.
and then we'll go to there. Maybe the next shift will be there, but we'll do a little more here, then head to here, and then head. I think that's a good plan.
Can you relay that to the next person?
Oh yeah, that'll be all of it,
Okay.
which I guess talk in a science meeting.
Okay.
And then the decision will be when to put it on also, depending on your coring needs. If you now want to core here, that may be another factor.
I don't know. I just woke up and trying to catch up, but Jeff said that there are nice terminal anomalies here, at least with the aerobicity. If temperature went up a little bit a few times, I think it may be worth to do one core on the side of this to see if there is an actual terminal anomaly and connect to the fact that maybe there are deeper fluid coming up, and that's gonna be it. If you do something, but it's gonna eat up in the time of the coring data.
I think at the science meeting, how much time we want to spend here.
Yeah.
At the moment, we're meant to come up by 10, which would mean leaving at 8.
Yeah.
But if we want to do more mapping or more sampling,
Yeah.
we might want to push
Right.
it. But then it depends on what you want to do.
Yeah. No, definitely. Okay. Few
20
minutes.
Okay. If you've watched along, this is our 17th dive, so we've been out here for weeks. Started up quite north of Valparaiso in a region called off El Quisco, the El Quisco Seeps, we call them, and that were wonderful, 400-meter seeps. Then moved south to Concepcion, more seeps with also a canyon along the way, and then another canyon off Concepcion, the Bio Canyon, and then came here to Isla Mocha. Just dived here for the first time yesterday. So we're much deeper now, but looking at this easy coring that's sort of happening, the coring in the past has been much more difficult. Even yesterday, the coring was a challenge, so this has been the easiest coring so far, except for out in open sediments. Yes. In about
Find a box, that's what I have to do there.
photo of all this?
Yes, please, Ben.
I have to watch him. Put
Yeah.
photo.
Yeah, go for it, whatever you need to do.
Should go photo.
Okay, wonderful. Let's hope they all come out nicely.
Mine's better done if you get the first three and already watch out, so we're not watching it now, right? You wanna do that?
What's that?
Maybe if you do the first three over there, or the hardest one, and already watch out, so we're not watching in and out all the time. It's easier.
Which one do you want me to get?
Or whichever you wanna get first and go back. So
Where you want to put them, in the back row, yeah?
Yeah.
Okay.
So I'm gonna watch out first, right?
Okay.
Let's see what happens. If you grab those three on the right, so you don't have to go to the left so
Sure.
much. Maybe a zip picture. Got a map. As far as we go, hold on. Do they share it? So we can share it. I didn't.
This may be our last dive. We will figure that out in a few hours. We've got tomorrow, but the activities haven't been planned. Then on the 3rd, we'll have to start making our way to Port Del Monte, where we'll finish up on November the 4th, and quickly do a changeover for the next leg. And some scientists will stay on board, others will mostly leave. And the journey will continue, sampling more canyons, seeps, and the hopefully amazing triple junction down south, where we hope that there will be hydrothermal vents. I think quite a lot of the people leaving will head off for exploring around Chile. I think they'll head off on November the 6th, so look for them dives again a few days after that. We may dive tomorrow, don't know quite yet. Obviously yesterday's site we didn't explore fully. Doing one dive on a site doesn't give you a lot of time. It's pretty...
That one's sliding now.
Can you shake it out? Thanks, Doug. Yeah.
That one didn't look very good going in, to be honest with you.
Okay, it's sort
I
of...
didn't really like that one.
Is that the one that kind of went in easy?
It did,
Yeah.
and it looked like it was pushing the mud down,
Right.
so.
Might have been
not
some...
much confidence in that one.
Might have been some gas underneath. Yeah.
Do you want to stick that Ken?
Yeah, you could take it again. Thanks. We'll redo it.
Yes.
But just
Also
not.
because we don't have any more toys.
Yeah, we have to take it again. Yeah. Maybe if, sorry, Doug, if you could shake it a little more.
Yeah, sure.
Thanks.
Yeah, we don't record it.
Just missed out.
Yeah.
How's that looking for you?
That's good.
Alrighty.
Thank you.
Yep.
And that's the picture.
Yeah, we'll take another grab just so we can update the map.
Yeah.
Yeah.
Release it.
Okay.
Come on.
That one looked a bit better there.
Get something else?
Get his updated picture for him.
Oh, you want to get another picture?
Oh, yes, please. And then Doug can pull them.
Yep.
Okay. That's great. Thanks, Ben. So it's just moved. Okay.
Okay.
You want to grab one, Scott?
You finished all your Halloween candy, Ben?
Never enough.
All right, so we're grabbing one of these and putting it back in.
Yeah, thanks, Scott. Any order you want.
Okay. Right to left.
We've got lots of fun little skeleton shrimp there hanging out. Too bad we didn't see them yesterday. And there's lots of tubes here, and the tubes have a very interesting little worms called amphirettids coming out. And hopefully we'll get a good close-up and show you those at some point.
Oh, it's slipping.
Hurry up. Make it.
Let's go over here.
Is it fun? Is that where you're going?
Yeah, because it's closer.
you sit. There you go.
Wow.
Perfect.
Wow.
It's gonna work. I'm teaching this one. Again, so I'm reducing, well, I got, can you reach that one? You got, 14? 14?
Yeah, no, I'm going, 13.
Oh, 13, I'm sorry.
Yeah, no, I think that's a good view. There's a little drift in the arm there. You see that?
Yeah.
This one sticks. We'll try to go back row. So far, so good.
It's professionally done. Go around. You can reach the others without me 14. Oh, you might be able to get 14.
That was huge. Okay. Yeah, we need to do a calibration sooner than later. And this dive going to 10, 11, 10, 10, 10.
You can do it in situ, right? You have to be up on surface.
The calibration.
Yeah.
I prefer doing this subsea personally.
Yeah.
Right out of room there, okay.
And go for black one.
I guess I'll go red just because it seems to
Wanna go there.
It seems to want to go there, yeah. The way my arm worked out.
I think we're gonna have to go 14 now.
Okay.
I don't think you can reach it.
So, we're gonna pause here for a second, Craig. We're just gonna do calibration on the arm. It only takes a minute, really. I just have to get the joints into a neutral position. And press a button on the controller. Everything will be better.
Okay. How often do you have to do that?
when the arm starts showing signs of drift, when we press the freeze button and the arm moves afterwards, that's kind of the indication that the commands and the position numbers aren't lining up correctly. The other part is I have to put in the security password. It's 1357. Even better. I don't know. Yeah. It's the one that's set by the factory, and no one ever changes it.
I still won't remember that. I did that a long time ago.
Okay, we have password set, level set. Now I can do. Set server. Set. Okay. Now, hopefully, when I open the jaw,
There you go.
it still did it.
That's good.
Just a little bit. All right. So sometimes you have to do it twice. Anyway, continuing on. There's no drift on the arm, at least, just the jaw.
Watching out.
Okay.
Yeah, it's short, right?
Getting out of the way.
Nice picture. Okay, you wanna use the, port arm for that?
Yeah, might as well. Yeah, I think that's better. It's definitely more responsive, that's for sure.
Nice hit picture. Cycle on it. Okay, ready? So basically, the last two, just watching. Some good bush cores.
So I stand corrected. We have not found the deepest seeps of Chile. Falco 2 and Sebastian found them back in June, north of here, off the Atacama Desert. They were over 2,800 meters down, so we're about 150 meters less than what they found seeps at.
There should be a sample shot if you can go back to it.
Great. We got this, we got this, we got them.
The banana blossom, the whitish.
The white one?
The whitish, the banana.
Yeah, there's another one there.
So we need to collect this.
We got one already.
Okay, okay.
We didn't see. We need to look. We took the
I
snails.
have a spot.
Okay,
Yeah.
we took 2 snails. Okay.
So it's that. Just the clams.
Double bubble.
What are these tubes coming out?
The tubes are feather duster worms, and we would like to get some of those. The acorn worm, I think we'll leave, but first we're going to pick up some clams. Is the next request.
Puppy, clams?
ones right in front of us? or-
Yes, that's fine, thanks, Scott. And they'll go in a bio box.
Kind of like bio box.
1, 2, or 3.
Let me draw in a little bit so you can see better.
Sure.
Where is the apparatus? Where did you see the apparatus?
Remember the island? It was there. I should. We have to go back there.
Okay. They'd like 5, please, Scott.
Good. Which one's your favorite?
Biggest one.
Ah, yeah. Careful what you wish for.
Ones that
It's
are
by
kind
your
of.
box.
I had to open 2 of those last night, and it's not something I want to do again.
Really hard to open?
No, there's just a lot of blood.
Oh.
That's why you had this bucket full of bright water.
It was
blood.
bright water, yeah, it really was.
It's one with an enemy on it.
Yeah. Maybe we can take on that
We-
one too.
We could take the anemone off, yeah.
Alright. So which one?
Really, your choice, Scott. Yeah, but
No, good.
as long as one of them is the anemone one.
Well, we have 5. so.
Yeah. There you can see the Physicamides have a fused siphon. So there's an inhalant and an exhalant siphon, so they're drawing in water to breathe. They don't really feed via inhaling water. You can see there this poor clam's already bleeding. They have an amazing amount of blood. The shells are kind of delicate, yeah.
I'm gonna go 1B.
Sure.
1B, yep, they can just pile up in there, please.
I'm tracking camera. Alright. 1B. There you go. Pick your place and both available, A or B.
And sit cam too, please.
Harder. Look at you zoom out on that. There you go. One, good plan. And then you close that,
Yeah, that'll make it easier. Work now. Camera in.
that? It was always so short?
Oh, anemone this time?
Yes, please.
Mhm.
Okay, ready for push.
Not much more to come up with. There you go. There's 3. Keep going. 31. Clear. Roger. Portion in. Carolina.
Do you want 5?
Yes, please. Is this number 5?
4.
4.
3 in the box.
Okay, clear.
Wonderful. Thanks so much, Scott.
Clear.
Thank you.
One more?
Yes, please.
Who gets to pick this one?
Mhm.
There's a lot to choose from, isn't there?
Got the lone wolf over here.
Pick a good one.
Yes.
This one's rather large. Big puppy.
Not big. Sorry, stretching the camera on you.
Oh, yeah.
Ooh, that is a big one. Holy cow.
Oh, that's the foot. So they have a foot that can extend out like a big tongue. They actually can be mobile, so if they run out of sulfide, the clams will go on a journey, or are able to go on a journey and look
There.
for another source of sulfide. So sometimes the poor things go journeying off into the wild, and it's a random walk, I think, for finding the sulfide. And they often don't. Now you can see it's pretty much retracted the foot into the shell.
Clear?
Nice.
Great, thank you, Scott.
That's good spot.
One correction.
Anna knows where there was another kind of clam, an acarax, so we'll just go and try and set down where that was. I think it was back
In the
behind us, where we are now, Anna?
yeah, depends. Where is the hydrates?
Back near the bubbles?
Yeah, near the bubble.
Okay. I think the bubbles are immediately behind us.
Because we just moved on the bush.
Why don't you come up and back up?
Yep, thanks.
My mind was saying.
Somebody just posted on the chat. Letter D. Thank you for the information.
Then the backup noise.
So we did some mapping of the seafloor after the plume had been found by the ship-based mapping system on our arrival, on our approach to Isla Mocha.
What you look?
We're backing up. just a little
The
bit.
bubble plume was then targeted for the dive, and then Sebastian did low pass mapping this morning and has given us a great idea of the topology down here. So great credit to the mapping team for getting us right on an amazing site, or a seep. And we did find the bubbles, and we're just going to turn around and go back there to see if there's another kind of clam that we might want to sample. And then we'll maybe do a little more sampling here. We did find a mound with clams and methane hydrate, clathrate, earlier today, and there's another mound nearby that we'll probably go and visit. There you can see some tracks in the sediment. That would be from clams moving around.
Is that mound over there?
Yeah, is that where the bubbles were?
Because
Oh.
can't be. Maybe it's another thing.
Could be something else.
Yeah.
Looks more like a mussel. I don't know. We're going to zoom in and have a look. Well, it could be a sea cucumber.
Green one.
It's a sea cucumber.
Oh, nice.
You want a closer look?
Yeah, please. Yeah, I'm pretty sure that's a sea cucumber.
Zoom in a little bit. It's got bubble float here. It's gonna be a partial zoom on landing.
Yeah, that's okay. We will not take it. It's a late Moni, unless we end up nearby, but we won't. We're going to look for the clam
Clams are
near the bubbles, and I'm a little lost.
We'll zoom in on, so there's double bubble and bubble curtain.
Okay, so we've got the hydrate. Okay.
Roger.
So if we can be looking.
It looks like from the number of targets right here, is that maybe where you did the gas type?
Yes.
Okay.
Yeah.
this one. So the aggregates should be here.
Okay.
Should be in front of us.
Yeah, if we can
2
look
meters in front of us now.
somewhere in here.
He's pointing to the ones we're gonna grab, Scott, so that's who he's pointing.
There should be a brown.
You in
It's
position
like
to grab those.
black.
We're looking for a blacky brown shell.
That look like the one that we. But here with the fish. This one and the fish. That was what we saw.
Okay,
Lisa.
I'm not sure that's alive.
Yeah, you were not sure either, but maybe the shell.
Okay, we'd like to look at that. Thanks, Levi.
Understood.
And there's a cute little eel pout that we can have a close look at too. Acarax are another kind of clam that also have symbiotic bacteria. They've got a shell, but it's covered with a proteinaceous brown matrix and little finger-like extensions. And often they're buried. So there's, that's an acarax there. That one could be alive.
Yes, that was too.
Yeah.
Alright.
Okay, we could sample that one,
Would you like to
please.
use the ground? Which one? The,
This brown shell there.
okay. You want to, Scott?
I think you could pick it up, Scott, and it could go in the same bio box as the clams.
Okay.
It's
Is it delicate?
it's kind of leathery. That's I guess a good way to describe it. It does have a shell inside though. But I guess treat it the same way you treated the clam.
Okay. I'm gonna reach out and just kind of do a test.
Yeah.
Might be too far away.
Yeah,
I'm
small
gonna
one.
I can see another
On the
one.
edge here,
Yeah.
before reach.
Roger. Yes, this little thing right here, next to the clam, dark in color.
That's doable.
Yeah, and it's got these little finger-like overlappings.
Is it alive or dead?
We'll see. It might close up a little more.
Is it zoom over here?
We've seen a lot this cruise, but always dead. So we
It looks good, doesn't it?
So acarax is in another group of clams called Solomyidae.
It's kind of resting on top of the.
Yeah, I still can't tell if it's alive. They have blue blood, quite different to the vesicomyid hemoglobin blood.
Alright, cameras retracted. Ready for porch operations? Go.
Did it just fall?
You're good.
You've still got it.
Camouflage in the mud. Took my off for 1 second.
So it's the same box, 1A.
Yep, 1A is good.
Roger. Alright, 1 coming up in there.
Nice.
Room for more of them?
There are 2 more, but we could zoom and have a look and see if we think they're alive. The first one's up here. I'm sure we'll have to move. Could zoom in on that one though. Thanks, Levi. Yeah, just in there, right next to the fish.
Alright, it's got this little looking color up here.
I think that one's dead. So the other one is over to the right.
You look at the right one?
Yeah, there's one over there.
There. Yeah, that one.
Okay.
And that one could be alive. Ah, pink scale worm there.
Yeah, the one that we saw this morning.
Seems very flat. Yeah, I think that might be a dead one.
Alright, is it a?
I think it's just the shell.
Okay.
Oh,
Let's
okay.
go.
Yes, thanks. Oh,
Oh,
actually, no,
No.
I think it.
Yeah, it did
they
look like.
had something.
Yes, it was alive.
Okay.
Oh. Yeah, we'll take it. It's still the.
It's there in the club.
Yeah. Fooled me.
Okay.
Camera in.
Same box, please.
Okay. One. Open there. No. Nice. Nice, that was the right one. We still have to try to reach the left one.
Okay, I think we're happy with grabbing.
2 is good.
Yes, thank you. Now a little suction time, please.
Alright.
And what do we do? We could go to 4, please, Levi.
Is that enough for you?
Potential bloodbath.
Index to drop 4, is that?
Yes, please.
Alright, we're on 4. What are we looking for?
Okay, first targets are over here. If you can reach, I hope. The little scale worms. There's 3 of them.
Yeah, okay.
Let me know when you want to.
Yeah, keep an eye on the hose, coming across the top of it. Push
Hose.
over there.
It's pretty hard for me to see.
That's a twist like that. We're good. There we go. All right, going in. Yep, perception. Suction on.
This is
Okay.
a gastropod.
Going?
Can you slurp it?
Okay.
Thank you.
Oh, I think I saw it going.
Yep, got it.
Okay, go to the next 2.
These scale worms are very commonly seen at seeps and vents. There's a lot of different kinds. And I'm working on these with Avery, one of my students, and she's done an amazing job on our understanding of their relationships. Great. We found very few of that group here off Chile. Ah, I think you got 1.
Okay. There's the other one.
Nice. Scott, there's a shell up here that seems to have a snail on it. If you could zoom in and we'll see.
Yeah.
That was some good eyes to pick that out.
It's Javier.
Javier? Okay.
Javier is the youngest.
Okay. If you can knock that off the easier said than done, I'm sure. It looks like a limpet kind. Not sure.
It's a shell.
Swing around so like it's there.
It's a shell. That's
Oh,
Oopsie. Okay. That's fine.
Can you zoom out?
I think you got it.
Yeah.
Yeah, I need to get that rock out of there.
Oh, right.
Alright, I'm gonna kill section?
Yeah, kill
Alright,
section.
section's off. I'm ready to turn section on at your mark. Nice.
Great.
Okay.
Next, please, Scott, there's a cluster of tubes.
Okay. Tube cluster.
Yeah, if you can try and gather those in, would be great.
We know in one section.
Ready?
Okay, coming on. Here you go.
They might go into the sediment a little bit.
Okay. These also might get stuck.
Oh, yeah. Hopefully they'll come apart.
Okay.
So we don't have to worry about stuff.
Levi found some worms in the tube the other day.
The clams are stuck in.
Oh, yeah, yeah.
Yeah,
They were
yeah.
useful.
Like there was
The
one
scale.
tube worm.
Yeah, we have to
get the rest of these?
Can we have a zoom in? I'll just see if I can ID what they are. There's a couple of tubey. I'm just trying to see this red down there.
It's max zoom.
Max. Okay. Okay, that's a spaghetti worm. We're looking for the feathery crown worms. Can we pull back and we'll look down? I did want in this shell. There's another scale worm and a brittle star. And then we'll look for these little plumes of feathers for our final slurping.
Okay. Same jar, eh?
Yes, that's fine. See, in the top of the valve there's a scale worm, and then underneath it is the brittle star. Actually, several brittle stars.
Here we go.
And there's a whole row of amphipods perching on the edge of the shell, like a flock of birds.
You want those?
Yeah, sure.
All right, going in.
Well, that's another idea. Yeah.
It's got-
Maybe we'll slurp. We'll leave that there and
Pick those
slurp
up.
elsewhere.
Use the shell as a speaker. We'll just pick those later.
Yeah. So if we could just look down here, please, Levi, and I'll see if there's another. I'm just looking for these little feather worms. More tubes. Yeah, these kind of things.
That's got.
Yeah.
There should be bigger ones, but that's the only one I see.
Yeah, we can just pan around a bit, please, Levi. And
Oh, there's one tiny one there.
Look, I got to
it's a nice snail. I'm sure we'll ask for the snail. So we'll get the snail. Yeah, these are the tubes. So
Doesn't look happy.
yeah, they pull in. Yeah, that's one that's in its tube there. So how about we go for that one, and then we'll go back for the snail.
See that one? What now? Good luck.
Yeah, I think you'll have to just push in among the clams, and hopefully the tube will come off. It's probably attached to one of the shells.
Can you point to it again?
That little brown tube coming out.
I don't know what section.
Yeah, suction.
Good. There you go.
Yeah, it's on the shell.
I wonder if you could take the manipulator and just grab the whole shell, tube on it.
Yeah, maybe we should just take the shell, and along with that other shell.
Oh, man.
Okay.
Yeah, it's definitely that.
It's attached to the shell.
He bumps that shell.
Yeah.
Alright, section off. It's
Okay.
the, see you, man.
All right, I think that'll work.
I was doing the challenge.
Do you want to stick with the gastropod or?
Yeah. Oh, sorry. There is the snail.
Here.
Yeah, over here.
Oh, it's got. See this now?
There's another challenge.
Ah, okay. All right.
Get you set up for it, and we'll go back in. It's not hard to see, but it's there. Nice. Alright, ready for session?
Oh, yeah.
Alright, here you go.
Wait. Yes.
Nice.
Thank you.
All right, that's quite a container.
Best container. All right, now we're doing
Now we'll grab a.
bucket pool.
Yes,
Alright,
please.
section off. There you go. What containers do you want these in?
We can put them into BB1A.
Alright, so this one delicately get this
There's six.
package.
There's 7.
Yeah, I think if you grab one of the valves, Scott, it should hang together. It's a beautiful scale worm there. They often have a very nice iridescence. I'm just counting the scales. 1, 2, 10, 11 pairs.
Oops. Or back.
Or you could close the valves and then no one will get away.
That ain't talking. That's good. That's what's good.
There's some seep.
Oh, look at the hydrate.
Oi, oi.
One brittle star has escaped.
Another 2.
Try to get some sit. Nice.
Oh,
Oh, I crushed
wow.
it. Shoot. Ready?
I think it's still going to work.
Alright, track the camera. Alright, torch camera. Routes. One opening.
Uh-huh.
Oh, sorry.
Okay.
Clear.
Scott, are you able to reach up here?
I think so.
So that clam there.
Oh, that other brown one?
Yeah, if you can.
Okay. And you still want the other clam that has the 2
And then we'll come back for the little one down in front. Thanks. Same buy box as before.
I can move closer, Scott. It's too far out.
There, yes.
Okay, maybe get the one in front of us first.
Yeah, that's right on the
Yeah, I'm just looking for the one with tubes on it. Maybe over here, actually. I can see one with the feather out.
Can you use the port arm to grab this one? See this one over here?
Okay.
Well,
Not the one that
I
had
feel
the
like
tube on it.
maybe when I drew it out, we modified one of these.
Yeah.
That we're looking at. It was a dead one.
I thought it was right here.
Unless it's a,
Yeah, so, I thought it was right here.
maybe it's right.
I mean, that's as far as I could reach with the suction hose, so it's gotta be somewhere in this area. But no, it's moved. Yeah, it's that right there.
See a dark dead one?
Yep. Because it had the two worm on there, on the upper edge too.
Okay.
Yeah, you could have. Here, I'll zoom out. So you can find yourself.
Geometry back on the worm.
Or you're reaching way up, Scott. You're reaching way up, Scott.
Couple of west nest up.
So I'm gonna zoom in slow, back in on it. Look where the dust clears. You gotta trim the nip can. It's that dark one there. And this dust cloud. We'll pull it out and we'll see what we got on it. Yep, this is the one I think it is.
Yes.
Yeah. Oh, it's twofer. One's a valve.
Flash Friday.
Alright, camera's in.
Okay. Same buy box,
Okay.
please.
Every time we draw in and out. There's much mud.
No.
Oh, on the porch.
He decided it was not Tuesday.
There you go. Alright.
Okay.
Do you want that one? Porch.
May as well, if it's easy to get, yeah. Great.
Off the rim, nice.
That battle box has a lot of good stuff in it.
Yeah.
Alright.
Last thing here is to bump forward a little bit and we'll go for that final clam up there.
Mine was the porch out.
I was gonna, I gotta get you closer though, so. I'll just put you. I'll hop closer, okay.
I could do a little coffee today.
So we're really trying to get a snapshot of the diversity of microbes, animals at this seep, to compare with seeps in other places, to find new species, to understand the ecology. And we've really probably only got this one visit to try and quickly gather as much as we can.
Alright. I think there's other fish coming up.
But we also want to go and explore, get a bit more of a sense of what is here. So we've got to, it's always this tension of staying and sampling and moving on, finding more things. But this dive's probably going to run another 5 hours or so before we leave the bottom, so, or maybe 4. So still plenty to look at. Yeah, we feel very privileged to be able to do this. Oh yeah, there's lots of tubes on that shell, so that has the feather worms that we're interested in getting. Yeah, that is definitely a dead one, but it's got some inside of the valves, so it has still a calcareous shell, but it's covered with that. It's called a periostracum, a protein coating.
Still want it?
Yeah.
There
We'll
are
take it for the tubes.
So the tubes, yeah.
Plus the shell would be useful for someone. They look so cool here in the water, but they're relatively hydrated, so when they dry out, they crinkle up and become very brittle.
Projection?
So this shell is called Acharax, A-C-H-A-R-A-X, if you want to look them up. So,
Yeah.
more Niscans?
Yeah, I think it's better because it looks like that a lot of bigger ones. It's
Okay.
better, and then we leave 5 and 6 for the other spots.
Okay, famous last words.
I hope so that.
I wish we kept a Niscan. I wish. Okay, but it's a pretty cool place, so we will take Niscan number 3 here. It's a bit dirty. Do you want to wait?
Yeah, wait a minute.
Yeah, we'll wait a few minutes. We are going to move, but we'll let the dust settle and take another Niscan here. Thanks. That was some really great collecting.
Yeah, one is for sure for the one that the for sure is fine. patchy because we also like background and like view background, so it's fine. But if someone else wants to water, it's better to close another one, otherwise I can't use.
Let's
much.
clear out
But for you, it's already.
And we
No.
can usually get less than 1.5 liters.
Yeah, but it's fine.
You say when, Anna?
Me? Just wait a bit.
So we sample water here. Because you can just see the bottom of a grey bottle there above the arm. We have a series of those. And 4 of them hold 5 litres of water and 2 of them hold 2.5 litres of water each. So each dive we can collect 30 litres of water. We usually do it at different sites, but this place is looking pretty interesting for several of the groups on board, so we're going to take our 3rd water sample here.
Greg, there's some interest in the chat about how you got to where you are today. Could you tell us a little bit about your career path while we're sitting here waiting for an biscuit?
Well, some might recognise from my accent that I'm Australian, and I did a marine biology degree in Australia. My interest was really in shallow water. Australia doesn't have much in the way of deep sea exploration technology. And I was quite happily exploring in Australia. Felt lucky enough to have a job doing that, but was invited by researchers to come to California with my background in biodiversity to study whale falls. And when I did my first ROV dive, that was really an eye-opener for me, studying whale falls. And got me hooked on deep sea biology, and a job came up at Scripps, which is part of University of California, San Diego, and I had a museum background, so my combination of biological research and museum expertise led me to be successful in getting that job, and so now I curate the benthic invertebrate collection at Scripps, as well as I'm a professor in marine biology there, and have been able to keep accessing deep sea opportunities provides with their amazing vessel. And we'll take these samples back, split them. I do the genetics work back at Scripps, and the samples will, the specimens themselves will go to the museum, National Museum in Santiago. So basically the requirements for me were to get several university degrees. And like carve a niche. I mean, my work is mostly on worms, so who would have guessed that knowing worms turns out to be a handy thing for exploring the deep sea?
How did you get into worms?
We do 1 year after our initial degree. And the professor I wanted to work with said, oh, well, you could work on crabs or you could work on these kind of worms. And I thought, having done a lab on marine biodiversity, that the worms were actually pretty cool. And so I chose that because crabs are kind of uniform in many ways. even though they look quite a different, they're not as diverse as worms. So that decision led to me continuing now many, many years later, still working on worms, as well as lots of other projects. I like echinoderms as well. And even the acorn worm we saw earlier, that's a research area. We study those. And but echinoderms and worms are most of my research time. Although Xenotabella, the amazing creature we saw earlier, which I hope we're about to see some more of, I've spent a lot of time working on those. But the key, I think, influence in me getting here was Bob Reinhoek at Monterey Bay Aquarium and Research Institute found a whale fall back in 2002, and that had on it an amazing worm eating the bones. So those bone-eating worms were the pivotal thing for me ending up being able to spend a lot of years being a deep-sea researcher. Okay, we are going to journey on.
Thank you for the
We'll cruise over to the next mound. I think that's got a waypoint on it. Along the way, we'll look at the clams, and there are the purple socks. You've seen the purple socks, Levi?
Maybe it wasn't here.
Okay,
Yeah.
how far out we zoom?
Looking for another mound.
We're going to head off. There's a mound. We'll go over this feature, heading towards this mound up here.
M3 Poi or Mark?
What inches is these
Yeah, I think it's this mark here. Yeah. But we'll go over the as it elevates up and see if there's any rocks. We're kind of interested in rocks. And we'll go slow. We might stop along the way. There's still we collected a couple of these purple socks, but we didn't get, we just want to get a few more images of the socks as we go along.
We keep calling socks.
You know, I did say socks as a joke.
We started calling them socks in 2008. Yeah.
Oh, amazing.
Yeah. The pilots called them the socks, the sock worms.
That is the technical term.
Yeah.
Scotty, can you tell us how you became an ROV pilot while we're transiting over to the next waypoint? Because there's so many people in the comments that are wondering how everybody got their career path. This is a serious question.
Okay, so I won't be sarcastic. Although that is my nature. I started out my career as a merchant marine. So I worked on break bulk carriers in the engine room, steamships, that kind of stuff. I was a steam engineer.
Steam.
And yeah, unlimited horsepower, steam engineer. And then from there, became a chief engineer on a diesel. And then from there, went shoreside. I was a port engineer. And then took a break from shipping. Was actually a bike messenger and worked at a bike shop in Washington, D.C. for a little bit, just to clear my head from engine room stuff. But then my wife at the time got a job at Woods Hole, and so we moved to Woods Hole, and one of the ROV pilots there was newly married and had a kid on the way and didn't want to go to sea anymore. Common theme. And so I applied for the position. They hired me as a contractor, and that was it. Since then, I've been ROV, and that was 2004 when I started at Woods Hole. And now I bounced around to many different ROV outfits, including Bari, Ocean Exploration Trusts, and some other independent gigs. One of them was Deepwater Horizon oil spill analysis, doing manganese nodules in the Clarion Clipperton zone, all of that, just independent contractor work, and other various companies, landed here in Bari. Was an ROV pilot there, and when they sold their vessel to Florida Institute of Oceanography, they're building a new one, and in the meantime, I'm an ROV pilot, so I was like, well, I kind of want to go drive an ROV, so Schmidt Ocean had a spot available, so I came over here. Yeah, so now I'm here. I hope that wasn't too long of a story, but that's my career path. That's how I got here. Yeah.
That's literally perfect. Thanks, Scott.
Thank you.
Yeah.
Yeah.
Through all
Nice. It's the most xenodeveloper I've ever seen.
These are all.
These are clams that are kind of making tracks. It's pretty nice to see how they move there.
On the bridge, that's an updated waypoint.
They're hunting for a better food source. Oh, is this microbial mat coming up?
Oh.
We're now going to have some gnashing of teeth for the lost push core opportunity. Can we drop a marker here? Just in case. But if we could get a nice zoom on this, Levi, it would be great.
So, how did it all work?
How does this
Target. Call it. What do you want to call it?
Just call it. Can we zoom on it? Is it a mat? I just want to.
Yeah,
I think it's a mat.
Alright,
Black
zoom in
mat.
further. I'll float here. Scott, zoom in on it.
Oh my God, too many things. Drop the target, zoom in.
Exploration.
Woohoo!
Yeah, you can call it black mat.
Black mat.
Thank you.
Teeming with life.
Yep, lots of amphipods there, copepods, a lot of crustaceans, little white scale worms.
2,700 meters below the surface. Let me keep going.
Yeah, you can pan around. We'll have a little more of a look.
More about that.
Peter, what do you think about this map?
So I was conferring with the group, and there's not actually a consensus that this is a map.
Ah,
Ooh.
what do you think it is?
On the bridge, there's an updated waypoint for
It
you.
looks a bit like a map. So, but there's some thought that perhaps, well, one, I mean, so for example,
Rocket.
what we just panned
Good
over,
afternoon.
that looks like a map to me. But
Good afternoon.
there's also some concern that at El Quisco, when we tried to map these, or sorry, to core these, it didn't work very well. So there is a bit of teeth gnashing, I'm sure. But we did get quite a few successful courses today.
That's quite a view of. Yeah, thanks. It's.
Oh, wow.
The
Yeah, that's nice.
That's very nice over there. Can we just hop up over here, please, Levi, and we'll have a look here because the caprellids on the mat is really cool, and I just want to see that seal.
Greg,
Yes.
there is a gastropod. There
Where?
was a gastropod there. Look, here. Down here.
Hang on. Well, we're going to sit down and we'll have another look.
They were white.
I think it's really interesting how you see these skeleton shrimp that appear to be grazing on whatever these features are, whether they're maps or not.
Yeah, caprellids normally feed on things that come by in the water column. But as we saw, there's a lot of other crustaceans there on the mat. And they may be preying on those, all the wandering copepods and other amphipods.
Oh, see.
Rather than going for the bacteria themselves.
Yeah, I agree.
Okay. There's a big snail, Anna.
Yeah.
Wow.
There was a sea urchin somewhere.
Oh, yeah, that's bad. I sat right back down.
Oh, we stayed where we were?
We stayed where we were.
Oh, I see.
The urchin's right up here.
Okay.
Might as well get the scoop going.
Yeah.
Okay. One. There's the gastropod. Let me see if I can get you a zoom in.
Oh, yeah, thanks.
Before we lose it. When I throw her out, it can.
Hey Eric, we're gonna do a scoop here, sorry, can we pause?
Okay, that's dead. Yeah, it's already a dead one.
Not interested in that one.
Well, it can be in the scoop. There is a live one over to the left. Okay, but we'll scoop we want to scoop the mat, like the surface of the mat, down, in, like just the first few centimeters.
Understood. Foreshadowing. When are you gonna reach them? Is that enough for you? We're getting the push off further.
Yeah, just grab and scoop one.
Grab and scoop one,
Yeah,
yeah.
that's fine. As I'm doing that though, just keep an eye on
The cords.
yeah.
This camera's gonna do that.
Come down on the shoulder.
Yeah, I could go up further and get you in the gap, too.
Or just use the other arm.
Use the-
I didn't. Usually I don't think I could use the other arm, that ram might be.
Gas tech man's pretty decent.
Yeah, okay.
Yeah, why don't I draw up more and get you past level 3 there?
Okay.
So we're going to scoop some of this sample of this
There you go. There you go, window.
putative
Look at that.
mat, and the goal would be to get a lot of the animals in there. If we wanted to get a better sense of the mat itself, we would push core, but we've used all 20 of our push cores today. We're still on this initial feature where the bubbling was. We've got another mound that we want to head towards, so we will continue forward in a little while. We've got sea urchins to look at. There's the jellyfish sitting on the bottom that were seen earlier today, and also hanging around here. They might be feeding on the bottom, which is kind of unusual. There are jellies, famously the upside down jellyfish that lies on its bell, tentacles up, and that one is using sunlight with symbiotic algae in its tissues. But here, no chance for that, so those jellies are doing something else. So Scott's going to scrape over the black parts.
Black marks. Yeah, is there like a particular one of interest? Or?
No, just any of the black, and don't try not to dig it in too deep, please. Just sort of parallel to the, dig it in about 1 centimeter or 2, and then.
Okay. Well, I'm
just-
gonna go for the target rich environment over here.
Yeah, that looks good.
Okay.
Perfect.
I see your technique, Scott. I see it. That little lift.
Did I have a
And
little
back.
A little bit of lift.
And then back down.
Yep.
You've been doing this for a while, huh? Shows.
Yeah, because most of the animals just live in that top layer. You get down 5 centimeters and it's no oxygen and no animals anymore.
Hmm.
So it's most bang for the buck is to get that surface.
You want another scrape?
Yes, please. Oh, look at that.
We caused something.
Okay.
Did I tell you there's a lot of methane here?
And that will do us. Thank you, Scott.
Okay.
Alright, so you can do one B.
Do you think it'll fit in BB2A?
Well, 2A already has one in it, doesn't it?
Can you fit both in there?
Maybe.
Yep.
Alright.
I mean, maybe I'm saying that prematurely, but I don't know how full.
Yeah, don't know what the
that
first
bag
one
is,
was.
yeah.
Alright, here we go.
We'll take a look.
It'd be nice not to put a bunch of.
If not,
sediments, that's-
put it in 2B next to it.
Yeah. Two. All the way in. Here we go. Here you go. Get away.
Dust to clear on this account. You know what, I think I'm just gonna let it go right there. Work
Yeah.
out.
Nice. Alright, that's
See if the lid squishes it in. Otherwise, okay. Okay, I'll poke. Oh, nope, squish.
Nice and close nicely, yeah.
Perfect.
Alright, we'll draw back in.
So, how about you open a quiver, please?
Okay.
And Levi, I think we can try and grab the snail,
Okay.
and then maybe we'll go get that urchin. Thanks.
I do think that snail is just a shell.
Oh, the one over here?
Yeah, I saw a
I
hole.
think it's just
there's holes in
shell.
the
They're
shell.
both dead? Okay.
Yeah, if it's
Yeah.
both that, that's it.
I think there's a hole in the top of it, right? Did
Ah.
you see that too, Heather? Yeah, there's a hole.
Oh, yeah, also dead.
Yeah.
Okay.
But I'll still open the quiver, I mean, I'm not trying to
Yeah,
get that.
no worries. Well, let's go look at the urchin anyway, and we might end up taking that, but we'll hold on the quiver. I'm just not sure if it's a new one. It looks like it's holding on something on top of itself, which is a behavior of some urchins.
Start there now.
It's got a clam on top of it.
No, aggregations on the top.
Yeah. But, that's the urchin we got yesterday, so we don't need it. Okay.
I know.
So do you still want me to open the quiver?
No, no thanks. We'll head on our way to the next mound.
Yeah.
Here we go. There you go.
No tube worms today, Levi.
No tube worms?
I'm shocked.
Yeah. Yeah, you wanted some more, right?
We did.
The segmented style?
Yeah. I figured we'd see them. But
Absolutely.
we got the purple socks instead. Actually, could we look at that one?
Oh, we got
That
the
one's got
purple
a
socks.
that one's got a posture that's kind of normal, but the way they crawl, so that'll give us a nice frame grab for the anatomy. There we go. So now we can see
Ah.
that's one's actually moving. They move pretty slowly. And can we, are you down or holding? Okay, if you can just hold there for a sec. So that's the dorsal surface, the mouth is underneath. And they hold their body with muscles to give it those folds.
Here's the mind killer.
Yeah. Yeah, he's hopping to the bearing 300, he's hopping to point of interest. Take your time all the way.
Oh, right now?
Yeah.
Okay.
Just slowly move to the point of interest. I got the heading
This
right.
is as slow as we can possibly do.
He wants to zoom in on this sock right here. I dusted it, these two socks.
Okay.
He wants to zoom tight, zoom in on those. Dusted it, we're waiting for it to clear.
Yeah, socks.
Zoom in on the socks.
Ooh,
That is another thing.
that's a hemichordate. Could be a different hemichordate actually. Mm. There we go.
Yeah.
So
Oh, it moved.
they do move.
He moved.
Yeah, that's how they normally crawl. The ones we've seen today have been like folded and dropped all over the place. So this one is how they move around. So their head's away from us.
Looks like somebody's ear.
You can see them moving.
UFC, just here.
Yep.
Yeah. Once I begged to have 30 minutes to just watch.
See how
I've
fast it
moved
moves?
about 50 centimeters and made it made a speed up version of it, and it did an S shape, so it was interesting that it followed an S.
So this, by other attempt.
If you could frame it up above the whole body, thanks, Doug, that's great. Okay, that's kind of the frame grab I want. And
Put it in the center of the screen.
yeah, that'll be perfect. Okay, that's good. Well, I did want an isopod, but it's moved on.
Greg, just a question.
Yes.
These belts that has on the top,
This, yep.
is determined part of the body or is just
The mouth is just in front of that underneath. We don't know the function of that girdle.
Okay.
It varies in shape. The animal can change it to bulge out, or I have lots of different pictures of how that girdle varies. Can we have the lasers on just for a sec? Thanks, Doug, and that'll give us a good scale. A bit over 10 centimeters. Yeah.
Okay.
You can carry on. Thank you. Oh, actually, we might slurp this now that I just saw this. That's a different kind of hemichordate.
Lots of meetings today, Doug. Am I still employed?
For now. All right, come out, starboard ram.
This we can go into 7.
Starboard ram's all the way out, indexing to 7.
Sorry. You want to take over? No, we moved the shifts.
Okay.
I started at 1.
Wonderful.
And what about the science meeting? Well, you'll have Amanda sit here?
Yeah, well, yeah, Amanda always has.
Okay, so you start at 4.
Yeah.
Okay, and you finish?
Yeah.
If we can get a really nice image of it first, and
Zoom in there, Scott.
yeah. Yeah, so this is different to the one we collected earlier. It's kind of got a darker head, and it's got these kind of wings on the side that we can see curling around, and then
There's a bunch of urchin spines.
yeah, there's a dead urchin it's sitting on, but it didn't do that.
Okay.
But then we can see the gut is full of sediment here, and that's where they tend to break.
Okay.
So they're very heavy at the back, and there's nothing up front.
Okay.
So, Scott, when you put the suction on at the head end, it'll gently lift off, and then eventually the sediment part should follow. But they do have a tendency to break
Okay.
in the middle. But
That pink guy inside the shell there.
Is shrimp?
is it a shrimp? Oh, it's a mysid. Yeah, it's a relative of a shrimp.
Ah, okay.
Yeah, nice.
Cool. All right.
the dark end. Yes,
Once you
please.
get there, I'm gonna zoom right in so I can go nice and slow
Okay.
with the suction.
Yep. Sounds good.
So these
Try to get it.
deep sea acorn worm group were not very well known. They were known from deep ocean photographs and some very poorly preserved. When you see how delicate this is, you can imagine how they come up with normal sampling methods. And it wasn't until ROV started sampling them that the anatomy was properly understood,
Slow
beautifully
down,
done.
dang it.
Slow it right down.
Wait.
And now quite a diversity of them have been found in particularly the eastern Pacific, but also in the Atlantic. And more species are being named. I'm just waiting to see if it comes through. And the Xenotobel is slowly making progress underneath us.
Nothing yet, huh?
I got it like on 1 or 2. I'm going really, really slow. Trying to keep it whole if I can.
When you first turn it on, there's a shell that popped in there.
Okay. Yeah, I see something 7 right there.
That's it. Got it.
All right, awesome.
Oh, and it's complete.
There it
Awesome.
is. So
Well done.
Greg,
All right.
I think one of the snails that we suctioned.
Yeah, it looks like it went in there. See it just at the bottom?
Oh, okay. Well,
Yeah.
with the snail.
Yeah.
Got it.
Okay.
I can't remember where. Oh, we had snails
It was in that,
in 4. I think.
yeah, it was
Wait.
supposed to go 4, but it
Right.
might have just gotten in the tube and then stayed in the tube. and
Got
fell
it.
down.
And look how much progress the Xenotobel made.
Anything else here?
No, thanks.
All right.
We
Go
can,
ahead, put that away.
we can head off. Everyone
Time
denigrates the
lapse that guy.
Xenotobel. Yeah, I couldn't tell if that was a male or a female. So it was probably a male. Usually the eggs are the most obvious things, big spots.
All right, you ready?
Yes, please.
There you go.
Are we making it to N3 fire?
Yeah, that's the. We'll head towards that mound. Would be great.
On the bridge, we can continue that move now.
Continue move.
So the seeps today have all been pretty much sediment, and we'd like to find some carbonate or rock if we could, as we'll get then another kind of animal bacterial community. You can see some tracks of the sycamines where they've been moving. They're big foot, they can dig down into the mud and glide along. So we've got a few targets from the mapping team this morning. Coming up now on another little mound. Can we just zoom on the black there? Doug? Sorry, it's a bit late. I think it's dead clam. Nice landing.
That's all the zoom right now.
Yeah. Well. Not quite sure what's going on there. Is that, what would you think this is, Peter?
I think that's a match. It looks like
Yeah.
the other one that was a little bit contentious about whether or not it was a match.
Yeah.
Like after your sediment scoop, we saw that nice sort of whitish layer at the top.
Yeah.
And to me, that's a dead indicator that it's a match.
Yeah, and for some reason we've just got a little patch of it here.
Yeah, it's probably, so these are likely sulfur oxidizing bacteria, so there's sulfide here. We know that there's sulfide because there's clams too.
Yep.
Right.
Okay, all right, thanks, Doug. I think we
Yeah.
can keep on going. And of course, there's a xenotabella curled up there. And a nice patch of live clams behind. And there's a sea mouse there on the left. Yeah, friendly sea mouse. We've seen those in the canyons.
Is that sea mouse or acarax?
Oh, it's an acarax. Sorry, Anna's right. I'm seeing sea mouse. Sea mice. Yeah, no, it's a dead acarax.
Oh, okay. All right.
Yeah, there's a few patches of mat here.
Thanks, boss.
Lots of Xenotabella in different colours. Really amazing how many there are. Acarax. They're dead. Acarax.
Yeah, basically different.
We collected 2 alive. So we're going up a little rise.
That is Senator the blue one on the left. We pass. It's okay.
Patches of clams are.
Yeah. An eel pout sitting there on the bottom.
Jeff, you'll notice up on the flight on the screen there, you're coming up on this depression here as well. It's a very neat picture.
Yeah, we've got an amazing map to work on here, and we've got a pretty strange looking feature coming up with lots of depressions. Can we just have a look at this, please, Doug? See if that's rock. I think we've got some rock. So we're at the top of this mound now, and there's all these sort of valleys down below us. So before we head down there, we'll just have a look around up here. It's not a great landing. What do you think? Is that covered in, those are sepulids. Sepulid tube worms. And they're a filter feeding group. No, haven't been found to be associated with bacteria, but they're relatives, the sabellid fan worms, which we were sampling earlier, have. So these are probably just filter feeding, but we'll have a close look tonight. There's an anemone, there's a lot of forams on this rock. There's a contracted anemone there. Okay, what do you think? Doug, do you think we could grab that?
Give it a poke and see.
Yeah, see if it's actually movable. Just before you do, actually, can you zoom? There's a baby Xenotabella and a big one. That's kind of quite a view.
Baby.
That's crazy. Yeah, that's really cool.
Baby.
That's all the zoom I got.
That's great. Okay, I love it. Okay, thank you.
Which part would you like to poke? In case it crumbles.
Well,
So lots of options there.
yeah, I guess nudge it at the base and see if it'll roll over. I suspect it's very fragile.
Yeah, with that view.
Yeah, actually, take a look up in the wrist cam. Kind of see the base is very rock looking.
Yeah.
And friable.
Yeah, I think that's too big.
It does look quite big. It's
It
got
goes
a
down.
big base on it down
Tip
here.
of the iceberg there.
Yeah, so maybe some to the left might be doable though.
Can you reach? Can you reach anywhere to go after this over? It's talking about like these on the left here. Probably that one right in front of the port arm.
Yeah, hopefully this one's.
But I don't know if you can reach that without smashing those push doors.
Yeah, the push doors are.
Sorry. I got a feeling they're gonna be pretty crunchy.
Yeah, that one has potential too, but yeah, these maybe.
It's gonna be a little bit crappy here. That was what was stuck on the bottom of the switchboard. All of us.
Go for this one first? Sure. Just because it's there?
Yeah, might as well. See what we can grab there. You know, if you need to see where it's gonna sit, right?
I think that's gonna be very fried. Very delicate. It's gonna crumble.
Yeah.
Certainly looks that way.
Try it.
This one here is our best bet, I think. I think it's our best bet, really. That one right there.
Try some more scoopage.
It's easier to turn around because
See what I mean?
when he just takes over, it's gonna do that.
That one's falling apart too. Real crumbly.
Yeah. Well, Lisa's wondering if maybe the big rock could be fractured somehow. I don't know if you've got reached over the back over there yet. Maybe if this would break off,
Okay.
I think she'd be happy.
Roll it towards us.
All right. Yeah.
Or if you can grab that horn and take it, you know what I mean? I'll zoom in for you. Center that up for you. That's pretty much it right there.
Oh,
Just fractured pieces.
wow, that could be fantastic.
I think that huge fragment.
All right, everyone hold their breath.
Yeah, don't breathe too hard though. All right.
Uh,
Come out on the
B1 or
first little slice.
B2.
Okay. Coming out real slow.
Okay. One has all the clams on it, in it.
Yeah, B2B or B1B, either of those.
Yeah, either B1B or B2B. Coming back with camera, coming out the porch. That's the porch all the way out. Two? Or one?
Your choice.
One, two. You're just about there.
I think that's going to be Lisa's favorite rock of the trip.
You're just about over it there.
It's kind of my favorite too right now, actually. I like it.
Amazing.
You're right over it, so if it falls, you're good.
Yeah.
I mean, you're
All right,
over the
everybody.
set, so you're on the set, so you're good.
So I think
Yeah.
I'll let it go.
All right.
That's fantastic. Well done, Scott.
Thank you. I wasn't being sarcastic, that actually was my
It
fun.
wasn't. I think it'll be an amazing rock
That was cool.
in the lab tonight. I'm signing off, everybody. We're handing over to the Chief Scientist, Eulogio Soto, and the Chileano, so enjoy the rest of the dive.
Thanks, Fred.
Hold on.
Shipping stuff, yeah, we work. They put it into where we are now, so new directives coming.
Remind Levi in about a half an hour.
And also, remember that you need to close the last 2 minutes before leaving on the bottom. But on
We're
the
gonna be
bottom,
working on this area
you
right
find
here.
other
We just got here,
okay,
so
otherwise close it.
if you wanna move a little closer, you can. There.
Yeah, when Amanda is.
Might be going there at some point. Hello. We just got here, so we haven't looked at any of this down here, anywhere, so you say where you wanna go. Okay?
You want to get there.
Okay, so that's the next spot up there, Ben?
Yeah.
Right up there. You wanna go up there?
Yeah, because there is a gap.
Okay.
Okay.
Yeah, you wanna go there?
What?
Yeah, okay. We can have that. No problem. All right, Ben. I'll just make my way that way.
Bridge updated position.
You should explore around here.
Okay.
After this,
Okay.
I would like to finish mapping.
Yeah, I know. Abhi told me.
You know, the project. You should see if there's any rocks here and tube bombs. See if there's any rocks and tube bombs here.
Okay. Muy buenas tardes. Saludamos a quienes nos están observando y siguiendo desde la transmisión vía streaming desde el barco oceanográfico Falcon 2 del Schmidt Ocean Institute de los Estados Unidos. Nos encontramos frente a la zona de Isla Mocha, explorando el fondo marino a 2.746 metros. En el marco de la expedición Cañones Submarinos, Filtraciones de Metano y Chimeneas Hidrotermales en el margen continental de Chile, centro-sur. Investigación oceanográfica multidisciplinaria liderada por la Universidad del Paraíso, la Universidad de Boston y la Universidad de Aveiro, en donde también participan científicos chilenos y extranjeros de la Universidad de Chile, de la Pontificia Universidad Católica del Paraíso y de la Universidad Católica del Norte. También científicos de la Universidad de Minnesota, de la Universidad del Estado de Oregon, de la Universidad de North en Noruega, de Insubria en Italia. Un grupo interdisciplinario, geofísicos, geólogos, microbiólogos, químicos marinos, biólogos marinos, oceanógrafos físicos y oceanógrafos. Nos hemos encontrado con la presencia de interesantes organismos asociados a los mantos de almejas ácarax, caliptogena, vesicomida, y con organismos no tan comunes como son los cenoturbelos y también como los balanoglossus o gusanos bellota, más comunes en profundidades mayores. Lo cual ha sido interesante poder verlos en torno a estos ecosistemas de filtraciones de metano y sedimentos reducidos, principalmente asociados a estas almejas que realizan quimiosíntesis bacteriana. Nos dirigimos hacia un punto de interés, principalmente por parte de los geólogos. Han observado una interesante fisura que quieren observar, quieren terminar de mapear. En la sala de control
se encuentra ahora Amanda Semler, de la Universidad de Boston. También está Yerko Castillo, de la Universidad del Paraíso. También se encuentra Maritza Castro, de la Universidad Católica del Norte, y también nos acompaña nuestro observador nacional, el señor Diego San Martín. Y también siempre con nosotros se encuentra a bordo un grupo de la BBC de Londres, quienes también están realizando filmaciones de hábitat y lugares de interés. Tenemos en la sala también la presencia de David de Oppo, de la Universidad de Lafayette, quien nos ha guiado para poder buscar este punto de interés, una fisura. Recordar que estos ecosistemas son puntuales, a veces son irregulares en su distribución, se pueden interrumpir cada cierto número de metros, son extensos, pero muchas veces son de distribución fragmentada.
Yeah, we just need to go to 25 meters above seafloor. When we are gonna map that square, so whenever it's good for you to go up to mapping elevation, yeah, that one is, and maybe a couple of classes just to feel.
So everybody has their samples now, except for the rocks. Could you
Okay.
spend a little bit of time?
Okay.
You passed a bunch
Okay.
in the last clam bed.
Yeah, okay. And we are
Um,
going to the place for interest for David.
To sample? Okay, after, can we go back and get rocks?
Yeah, we
I
just-
didn't get my samples yet.
We just need to map that square, because that's a very good feature. And it's gonna take 5 minutes. Promise.
No, just, we managed to get every other sample on the list today, except for the rocks.
It's gonna be nice. It's gonna be nice.
Maybe there'll be some up here.
That's it. Because this looks very, very interesting, this feature here.
Okay, but I didn't get my samples yesterday either, so I.
Yeah, I mean, everything is really organized in the watch that you're gonna charge.
No, I
What's
know.
going on?
It's not your, I'm just talking to the watch here.
Yeah, sounds good.
Yesterday we didn't get the rocks either, so
Mhm.
got two.
Okay.
So you get 5 every course.
Okay.
So yeah, try to
Yeah.
spend some time. You still have
I'm at
lots of
25
time,
meters.
but you can
I'm
go
ready
back.
whenever you-
Okay.
I'm just gonna run
Like the site
just
where
gonna
you just picked
free
up.
fly these lines.
At the same
Okay.
Where
place.
Greg picked up the
Okay.
rock, there are
Okay.
other rocks.
Okay. Okay. The same one.
The
Same
same
place.
site.
Okay, we're
Yeah.
recording.
Okay.
Okay, star lines.
Or if you see up here,
Ah, yeah.
it's fine.
Okay.
And you're just gonna go to basically where our turn.
Roger that.
Yeah, yeah, just to feel the square. Because
Thanks, Alan.
No, that's fine.
No worries, I don't want to run you down.
Appreciate that.
Como mencionamos, estamos en la búsqueda de un sector que no se alcanzó a mapear, que es de interés para uno de los geólogos, para David Oppo. Es una zona fracturada, un promontorio con un pequeño montículo, una pequeña colina bajo el fondo del mar, que presenta una fractura en su zona central, que podría presentar tal vez posibles burbujas de metano y tal vez algunas comunidades de interés.
Yeah, we can come to an old stop now.
Mencionar que dentro del fondo marino también hay zonas rocosas que son de interés para algunos científicos, algunos de ellos requieren la recolección de rocas justamente para estudiar la fauna que vive sobre esa roca. Es muy importante también el estudio de esa fauna porque la roca provee un sustrato, un refugio para estos pequeños organismos.
Hey, you wanna start from here?
it's okay.
Okay, we're just filling in that square gap.
That's
Okay,
it,
no
that's
worries.
it. It's a five
Okay,
dimensional.
yep.
So our swamp is about 25 meters to the side, so if you run up a line and go over 2 more squares and then just go
Okay,
down that, it should
that'll
be
work.
fine.
Okay, cool.
Yeah.
I'll run up this line that's closest to me then.
Okay, it doesn't have to be perfect.
Yeah, no worries. And you don't care if I make good speed, right? I could go like a knot.
0.4 is fine. That's what we're
Okay.
normally running.
Alright.
Davide, is this another potential hydrate mound?
Yeah.
Yeah.
We think it's very worth fill up the little square.
Yeah.
We're going to map now, or we are?
5 minutes. Yeah,
Alright.
just because this morning we didn't map that little square, so we want to fill that because that's most likely another hydrate mound. So if we finish the square here, you just drop to the bottom, look what it is. I think you will find more stuff, and then go back rocks for example.
This one? Oh, yeah. Right here. If you
Yeah.
just run down, that should cover it. Okay, end of line, you can turn. Free
Turning.
flying the mapping, nice.
And I can get rid of a lot of this dirt.
Yeah. Rinse cycle.
A rinse.
A rinse. A rinse. Okay, starting line.
Okay, going forward.
It'll be 3 hours to get up, so 10 o'clock.
You came at a great time.
This is exciting.
This is exciting. sorry.
We love this.
Because
I
we're gonna
missed- I
scream
missed that, but-
bubbles.
Yeah, I have a video of what happened.
I mean, I'm surprised that there is so many people here. What is the purpose of your presence?
Okay, end of line.
Turning.
And then you can just
Yeah,
head
that's exciting.
back south, Doug, and then we'll be done.
Well, let me know you're gonna burn something. El video está listo para que lo veas cuando tengas chance. Ahora tienes que estar aquí. Okay.
Okay, start a line.
Perfecto, ¿puede la reunión? Ok, perfecto, para ver si puedo subirlo y enviarlo a que lo revisen ellos también antes de, pero está bien, después de la reunión, está bien, sí, sí. What's that?
I know.
Yeah, you're so lucky.
Just a 2-hour job.
Yes.
We are done, so if you want to drop to the,
You ready?
to the bottom right now, yeah,
Hold on, not
2
yet.
seconds, but
Okay.
we are done. You may want to drop at the bottom here, and so just go back wherever
Okay.
you wanted to go.
So-
Flying close. So we can actually see what's happening.
Can we come back now?
I'll get that mirror.
I just want to get a little bit of overlap.
Yeah, I have to let the back end of the vehicle go past the green.
Bueno, hemos logrado llegar al punto de interés para uno de nuestros científicos que necesitaba mapear esa área y ahora vamos a retornar al lugar donde estábamos obteniendo las muestras de roca. Así que después del mapeo realizado, volveremos. Durante las inmersiones del ROV surgen siempre intereses de algunos científicos con respecto a visitar algunos lugares o recolectar algunos organismos en particular. Para ello es vital la coordinación con los pilotos del ROV, quienes nos otorgan las facilidades para poder ir a los lugares que nosotros deseamos, recolectar las muestras que nos interesan, poder estudiar los sectores que también son más importantes.
Got a feeling that square perfect. I respect it.
Nothing is exciting.
Down, down.
They could have been smiling at
Coming down to the seafloor.
So, you remember the place?
What place?
With the rock?
You too. No.
You follow my track back.
The previous place, the idea is to set up to the same place or around the same place. No, it's not necessarily the same place.
Well, we go back the same
Well,
way.
Yeah.
We discussed it, that you need to do that.
Gotta turn around and go back the other way.
Where we collected the last rock.
No, really, I'm doing all the tasks I needed to do in the last 3 weeks. I haven't done.
All this up here, so it's not close to the seep bed. And then you'll have to start moving the vessel back to where we came from again. Yeah, well, until we find rocks, so just go up south and I'll make my way back towards, see how the white line, my trail, kind of try to follow that back to see if we see these rocks. Yeah. You can start moving the vessel whenever.
Bridge, updated position. I think that's all.
Still reading 23. Yeah, good luck.
Pelagic sea cucumber.
Yeah.
Really cute. Feeding on the ground.
Somebody will want that. Gotta be quick.
So we are now getting again to the seafloor to continue the mapping on deep sea biodiversity from Mocha Island, Seeps at 2,750 meters deep.
It's that one that were hydrates, but it's just
Snap. Sure. Shell. Shell. Vemos un fondo, todos estos montículos de arena corresponden a foraminíferos aglutinados. También se observan pepinos de mar, algunos translúcidos, como una forma de adaptarse a la ausencia de luz y también a la presión. Todos estos montículos de arena corresponden a animales. Estos foraminíferos son invertebrados coloniales que se forman justamente, son protozoarios y se forman justamente ayudados a través de lo que son los granos de arena. Forman una colonia, un grupo de individuos y crecen de forma arborescente muchas veces o de forma aglutinada o agrupada. Voy a dejar con ustedes a Amanda Semler, microbióloga recién doctorada de la Universidad de Boston, quien tomará la dirección de las imágenes y del control acá en la sala donde estamos realizando la coordinación con el ROP.
Hello. How's it going, everyone? I'm Amanda, temporary science watch lead, permanent pushcore enthusiast.
You're doing great. With your support, I thought I would help.
What are we talking about?
All right. Sounds like main goal is rocks.
If the rocks are too big, they're very easily, and you could just take
What am I?
half.
Okay, great.
So
rocks
a
and
piece
throw
like
it
that
together.
is too small, really,
Okay.
but
So we're looking for a sizable chunk.
that would fit in there. But something like that could be broken.
Okay.
You know.
Should we pick this one?
Yes.
Great. Okay.
There's, I don't know how many, there's 3 empty biomass.
Yeah, there's
So it would
3.
be great to get 3 rocks.
Okay, great. Sounds like this one is reserved for pelagic, and then maybe people can help me decide what to put in there.
Yeah. If we see any corals, but
Yeah.
we don't have to fill it all,
Okay.
but we still have a couple more hours in the dive.
Okay.
So. But it would be lovely to get some rocks
We'll-
here, so.
we'll get you some
Even
out.
like, if you don't see them in your field of view, travel around a little bit and look. Because I think they're the last main sampling thing of them.
Will do. All right, can we zoom in on this guy a little bit?
Interested in the critters?
I think Lisa's interested in, yeah, the critters on the rock and the ones living inside the rock.
Could be like stone.
So it looks like this one's gonna be a little big to entirely put in the box, but maybe we can get a chunk off of it. Does that sound okay?
Is rock, man?
Not possible. And I think it's almost time for Chile hour.
Amanda, how's your science processing been going, like your sample processing?
It's been good. It's been good. We've gotten a lot of mud. So we're trying to, I'm on the microbiology push core team. So one of the things we're trying to do is figure out the growth rate of the microorganisms and the rate at which they oxidize methane. So both of those two things. And we can find that by bringing the sediment that the pilots are kindly collecting for us. We shove it in a bunch of gas-tight serum bottles. We feed them a bunch of methane to purge all the oxygen out, and then we can measure their growth rate over time. And it's been going well. Unfortunately, we don't get a lot of on-ship feedback. We have to wait for lab analyses when we go back. But everything's been working out, so we're pleased.
You're touching the core. You're touching the core. And you're not touching the core. You're touching the core again. Can you there you go. Squish in your.
Ooh,
That's a piece of it.
that's a good chunk.
You wanna look at it?
Yeah, let's do that. Oh,
Sorry, that was me. What's on it though? That's interesting. It's got a lot of interesting creatures on it.
yeah.
Kind of has like this.
Looks like some worms, worms maybe. That's nice.
Okay.
Ooh, that's a good one.
Camera in.
Right. So we've got 3A, 3B, and 1B empty, according to my
All right.
sheet over here.
You want to do a little bit of service or you want to get it in your okay? Okay.
Nice.
Let me remember your porch out. That shoulder on that one's too tight.
We gotta get into one. And your shoulder left a little bit. Yep. Stay there. You're gonna make it. It's gonna work. Yep. No, lids up. I'm covered. Yeah. Move the wrist down. Yeah. There you go.
Oh, nice.
It's in. You're in the right box. Nice.
All right. Thanks, Ben.
Shoulder left a little bit. Yeah, hold that. Don't come back right anymore. Okay. Alright, I'm gonna get that porch coming back in now. Are we looking at more rocks here? Good. Now what?
All right, maybe we just do a survey around here for like 3 minutes and see if we can find another good rock to pick up for Lisa.
That looks like a live version of the thing we picked up earlier today.
I'm not sure if it's
species?
alive.
Oh, I see.
Maybe we head south a little bit?
Hey Amanda, guess what?
What?
There's a really good question in the chat for you. How do you incubate the microbes? Do you pressurize them?
Oh my
This
gosh.
is from Jessica. She used to be she sailed with us earlier this year.
Amazing. Yeah, so we do not pressurize them when we incubate them. It can be dangerous to pressurize glass bottles to much over maybe double atmospheric pressure. So unfortunately, we're not able to recreate the pressure conditions. That's definitely a big limitation of our work, especially when it comes to measuring gas oxidation rates. But it's the best we can do for now. Our normal process, once we get the samples up on board, is to mix the sediment with water that we collect nearby. That's part of the reason you see us collecting Niskin water at these locations. So we'll mix them together into a sort of slurry. We'll purge them with nitrogen gas just to get all the oxygen out, and then we add a bunch of methane to the headspace so they have something to eat. That's the general process.
Eat of methane. You guys brought methane with you? Bottles of methane?
Pardon?
You brought bottles of methane with
We
you?
did. We have 3 big gas cylinders on board: an argon tank, a nitrogen tank, and a methane tank. So that was fun to try to secure on the boat. It's definitely something the MTs were concerned about, but we got them strapped in, so we're all safe and sound.
What are you going to use the argon for? If you're I'm not sure. Are you the one using it?
Yeah, we're the ones using that too. Yeah.
What are you using the argon for?
Yeah, we have a couple different uses for that. We're preserving a little bit of the sediment to take with us home to set up incubations back in our lab. And so some of that is to just flush oxygen out of these little bags that we store the mud in. We also use it to get geochemistry from these sediments. We'll take packets of sediment, put it in a piece of equipment called a pore water squeezer, and we'll pump a bunch of argon gas through it so that it kind of comes through a filter, generates a vacuum, and we can extract filtered pore water for a variety of geochemical analyses. So, yeah, we're happy to have the gas tanks on board.
And there's another question. Are you going to take samples from Isla Mocha?
Yes, the shallow, the very shallow site. So I'm part of the team that's going hopefully on land tomorrow. And we're gonna cross the island, and there's an intertidal methane seep site that we're gonna go visit. And those ones are really cool. There are videos online of these seeps. People have been able to set the water on fire. There's so much methane in the water right at the surface. So there's a lot of methane down here at these seeps, obviously, where we're measuring a lot. But they have kind of, the 2,700 meters of water above them for microorganisms and other organisms in the water column to remove the methane before it hits the surface. So most of this methane is never going to make it to the atmosphere, and that's not the case with these really shallow sites. So they're really cool.
Keep going.
I'm sure we'll find it.
They're right here.
There you go.
We're talking about some snails.
Wow. All of them. So I'm hearing this. Yeah. Do you think a suction sampler would be best for that? Let's see. Actually, I only have the little ones left. Can they go in the 540 micron suction sampler, or do we need a coral clipper?
In success sample is okay.
Even the fine mesh?
Fine.
Is that fine?
Oh, yeah,
Okay.
they are.
1 and 2. Yeah.
Yeah,
1 and 2.
1 and 2. Great.
Those are really small too.
Okay.
That'll be good.
shrimp are actually amphipods, right? Maybe? Propeller shrimp or amphipods?
What do you?
Because I remember they were in.
Close up. Partial section. I think they're all right here.
That's 1, 2, 3.
I see at least 3.
I see, there's one down here, there's one here. I think that's one.
Let's look. Pretty small.
Yeah, they're small. Oh, sponge.
Turn it on there.
Okay, it's off. You should just pick that whole rock up.
Yeah.
You want to pick the whole rock up? That's an option.
Yes, although if we
And
pick
then
the
just
rock
the whole
up.
part too, though. You can see how big the rock is?
Yeah.
You can pick it up, but it might, the whole rock might just come out. So maybe we do this. Maybe we start here. Get this going.
Yeah, I think let's do that.
We can get the rock for Lisa.
Ben,
Maybe give
they're
her
talking about picking the
samples
rock.
from.
You see when you
I
do
did
that?
this snail, but it's only if it's possible collect.
Because they can pick the rock up, but they need the space for the rock for Lisa. And so she can have the rock, and you can have the snails on the rock.
Only possible collect.
Oh, here we go.
Oh.
Yeah, we're. This other one? Yeah, click the one right there. We'll keep trying it.
Oh, there that one went.
There's this one.
There's at least 2.
There's one. You gotta get the picture. It's moving. You get it to budge.
I love the suction sampler. I feel like it helps
There you
get
go.
off
Yeah,
the
Okay.
There's one more.
fuzz.
Yeah.
See one more lower.
Get everything in one go.
Did someone say what this thing is?
Yeah, I don't know.
It's like the praying mantis of the sea.
Yeah, it's the amphipod skeleton shrimp thing.
Yeah,
Probably.
it's so cool.
Grabs the ground with its arms. What? Okay, yeah.
It's so
Inchworms.
wild.
These praying mantis arms, but also these antennae.
Shit in my helmet.
Interesting. Do you want to try to?
There was a 4th one, I think. We saw 4 of them.
There's one here,
Right there. Look at her pointer. Right there.
but it's kind of stuck.
Still inhaling here.
Oh,
There you go.
there it goes. Wow.
Right.
Isn't that one on top? Let's just stay on 2. See it?
There's a tiny one. Barely.
And the one in the corner, behind the white.
Oh, I don't know.
It's okay.
She said
Is that enough?
It's okay, thank you.
So
Cool.
that's 4 pedazos. 3.
3.
3 pedazos.
Javier is so happy.
Aw.
Cool.
All right, what's next?
All right, let's keep moving.
Okay.
That was jar 4? What was that?
Jar 1, I
Jar
think.
1.
Jar 1.
I think Lisa needs the rock.
She wants the rock that we just suctioned?
Yeah.
Okay. Sure.
All right, Ben, go for seconds. It's crumbly. Which arm are you gonna go with? Stop. Okay.
Where are we gonna put that?
You gotta go to your armpit, over here on 3.
Oh, I'm on airplane mode. That's why. I didn't know Wi-Fi. I'm like, I'm not getting any of these messages. Okay, great. Yes.
I got it.
Barely have it.
Nice.
Okay. Any more chalk?
Either of the 3s.
Okay, hold on.
How's my shoulder?
Oh, that's fine. Alright, I'm watching it and I'm coming out. Let me open it before you come in and go. I said 3.
Can you just watch my shoulder?
I better watch my shoulder.
Looks like we have one of those skeleton shrimp on this rock.
Alright, that's what you get.
One of the scientists on board, Lisa Levin, is doing counts of the macrofauna in and around these rocks. So she'll pick a few off tonight, leave it in water overnight, wait for the rest to crawl out, and then tomorrow morning we'll have a nice count of everything living here.
lot of stuff on it.
That had a lot of stuff on it.
What's your name?
All right. Let's keep moving. So we're seeing a lot of live clams at this site. Unfortunately, we had a dissolved methane sensor on board the last few dives, and we had to remove it for this dive because it's so deep. We're just worried about a malfunction and want to make sure we have it for the rest of the expedition. So unfortunately, we don't have real-time methane data on board today, but I'm very curious what it would be.
That rock's too big.
That rock's Why is it fuzzy? Can we zoom in on that?
There's a sponge or
Oh,
fishing net to the right.
yeah.
Can you zoom in on the surface of the rock, top edge of the rock?
Yeah, please.
Did they all close up? I was watching.
Not all of them.
You might pop wash. Interesting, the parts that are dirty and parts of the rock are clean. Must be the steep sections of the rock
Yeah.
where the sediment.
So it seems like different organisms are the white kind of worms are keeping it clean somehow.
What's off to the right? Is it like fishing net or is it like sponge? Sponge. Some sort of spongy business. Okay. So we keep going southbound? Shall we keep going southbound?
It's not two men then.
Yeah.
Just a driver.
Just make the bunk move. Just kidding.
I'll move forth. There we go. Where are we going back?
This is pretty dense.
Yeah. Oh, can we zoom in on the middle of that? Look at this portion here.
Get close.
Holy cow. Can we get a sample shot here? Not a sample shot. Observation.
Oh, it's really blown away. It's just the.
Is that all bacterial then?
Looks like it. Wow.
Take our heading off maybe.
Oh, look at those. Just tiny little snails. Wow, the zoom on this camera is so good.
Slave sticks, man sticks.
Wow.
Taking the field.
That's a lot.
Laborers.
Can you hold it for just a second? I wanna mark this. Show interest.
Wow.
It's like the tree people. No, yeah. That's not Game of Thrones.
Skeleton shrimp. They're amphipods. The praying mantis of the sea.
Great. Ants, yeah.
There's so many.
They're still in the animals too.
Oh, wow.
Very
cool.
That bacterial mansions. Yeah.
It's completely covered down.
I'm wondering if there's a spot, like, is this part a sponge right in the front?
I think it's bacterial matting.
This is definitely all mat.
Yeah.
Some of these more lumpy bits. Wow. Okay. Very cool.
You want to stay here and sample something, or you want to take off?
Let's take off. Yeah.
All right. Here we go. Come wide a little.
Jeez.
Bridge, can we move back 20 meters south, directly south?
We can be, and the question is, should we?
Yes.
Scientists said yes.
Okay, well, we shall. Science.
Thank you.
Thanks, science.
He said welcome.
Oh, here's another of those purple sock creatures.
Where?
Here in the corner on the left. Xenoturbella. It looks like someone's old shirt. It's like.
Yeah, I would not guess that this is an animal. But if he is in some.
Yeah.
Crazy how dense they are. They're all vertical.
Yeah.
So many.
Can we mark this really thick clam bed somehow? It sounds like we might be coming back at a later date.
Oh, we're coming back.
Come back.
Huh?
When do you get back?
6 o'clock when we normally come back, I guess.
It should be on with mine.
Where we were headed southbound.
Okay.
You got a good marker, Ben? Can you just label that, large clam bed? Thank you. Is that good enough description?
That
for you,
sounds
man?
great. Thanks. Okay, this belongs here. Just get all my effects here. One more rock that we should collect for Lisa.
She needs one more.
She needs one more. There's like biobox 3B.
Okay.
And we're seeing just a lot of clam beds and interesting things. It's a good dive. It's always a good dive.
Best dive ever.
So we keep finding these really thick clam beds and mats and things on top of these mounds. We think that one of the reasons for that is that hydrate is less dense than sediment around it, and so it kind of ends up floating and pushing the sediment around it up, almost like a raft or iceberg or something.
We've been around here, we've been around there. We went up there, we only did a little mapping and then came back. We never even looked in there, so I don't know if you wanna, where you would like to go.
Well, recently we were here.
Yeah, we've been around here, this is where we were, locating and picking up rocks, I believe.
We can go to the white point 15.
You wanna go towards that direction? There's not really much over there, over here.
15.
So how many rocks did you get? We've gotten 2.
You see on
We have room for
beds,
one more,
you can
and we
see
got
the
some.
map is better. You
With
wanna
the
go towards
snails that
there.
were
See where he's at? It's
Okay.
kind of flat. That's where
I
you wanna
know
go.
Still
they
a bunch of
wanna
coral
come
quarters.
up early.
Yeah, and you see up here in between.
Because we have the next dive's at 2 a.m.
Between 18 and 19,
So,
where we went
and it
straight
ends
up,
at
there's
2
like a
p.m.
little
So-
cut in there. I don't know if you wanna see that, or if you wanna.
Get one more.
Yeah, I know. Okay, yeah.
And also, this is such an amazing clam bed that anything you can do with lasers or whatever to figure out its dimensions would be really excellent. Put in so that you can estimate biomass or, do something before you leave here. What time, we're gonna come early, right? Because the dive is at 2 a.m. The next dive, so.
Yeah. No, I go for dinner at 6.
But what time are we gonna see? The discussion was to come up early.
All I see is clams, as far as the eye can see. You saw rock?
Leaving at 8 is the morning. You'll be here at 10.
This little guy here?
Yeah.
Yeah.
Yeah. That's arrival at 10 p.m.
7:30.
And the next dive is 2 a.m. You've got that. So all your cores, everything has to be back.
How's that so?
Stunning.
7. Are they doing any more mapping?
So is that here and here?
Geological? ¿Qué era lo que había en un museo? Había como una cosa redonda así.
That's not a rock.
Is that a different
It's a
kind of
clam.
clam?
I picked up 2 of those earlier.
You got some, so
Okay.
yeah, they're really cool. They're they had stuff like that, so that's so I always just found it square out. If once we leave the Carbonate area, I suspect there will be
That's so funny. I said it. When I looked up and we were zooming in on that.
Yeah, I know.
I'm gonna have to do this.
I know.
Really? Really?
I know. The cores are all in. We need to look. Are you traveling?
Nope. Southeast. Do you want me to travel north?
I was asking you about
No, we follow to the south.
I'm just trying to find rocks, so I'm just kind of going back until somebody tells
Also,
me.
if you get there at the triple junction, like that's good for you too. Like you, that's your thing.
And I was just talking about
How much work, but it's
There too.
Yeah, I know, but I just collected some, like, so, not you, but whoever was collected somehow too many minutes ago, so I don't know. What's the spot you got the last drop?
This was, oh gosh. There was a clam bed just before the big one that you came in here for. That was a little bit smaller. The coordinates will be on sea log, but similar depth.
you can pull them out once you log.
See, I can do that.
Next to this, next to the big one there.
Good work.
Only where, man?
Keep turning. Going back to the same place.
Blade. You sure?
It's going.
Oh, you're right.
Is that our cloud?
Yeah.
Sample geology. Let's see what's 38.
Sorry.
No shallow. Yes, shallow.
So, do a gravity clean.
Okay.
While you guys are all processing samples. Yes.
Okay.
Yeah, it's time.
That's the backside of that load.
Nice.
Here. So, this is rock exposed. To the right? Okay.
I thought there wasn't a board on your site.
So I can get ours back. I'm not gonna leave it.
You're talking about that?
Yes.
Yeah, I don't know. Half of me is on
Think so.
Clams. Yeah.
That one is okay?
Perfect.
Yeah.
See my very intentional. You see that in the 4K highlight, you know, you did.
Buenas tardes. Les comentamos que estamos ya terminando nuestro dive de hoy, que comenzó a más de 2.700 metros de profundidad en una filtración de metano muy diversa, con muchos organismos y con muchas formaciones rocosas interesantes que han permitido la presencia de muchos organismos que no habíamos observado en ocasiones anteriores, ni tampoco en los sitios que hemos estado observando durante este crucero. Durante el día de mañana tendremos los últimos dive en el sector de Isla Mocha para finalmente terminar nuestro crucero y emprender rumbo a Puerto Montt para regalar el día 4 de noviembre en la tarde. Este crucero comenzó el día 11 de octubre desde Valparaíso y nos dirigimos hacia la desembocadura del río Limarí, donde realizamos nuestra primera inmersión estudiando y explorando el cañón del río Limarí. Posteriormente estuvimos en el sector del Quisco, donde estudiamos una filtración de metano también muy interesante, y el cañón del río Maipo. Después estuvimos en la zona de Concepción, donde también pudimos registrar, explorar una filtración de metano en esa zona, y el cañón del río Biobío. Y ya finalizando, nos hemos concentrado en la zona de la Isla Mocha, también con el hallazgo muy interesante de haber podido encontrar esta filtración muy diversa, biológicamente muy interesante, geológicamente también muy interesante. Así estaríamos dando mañana con las últimas inversiones termino a este crucero que ha sido muy exitoso, muy interesante. Se han podido registrar los primeros hallazgos de ecosistemas de filtraciones de metano en el margen continental de Chile. Estamos muy satisfechos, llevamos mucha información, llevamos a casa también mucho trabajo. Hace un rato tuvimos una reunión con la gente del Schmidt Ocean Institute y pudimos concluir que ha sido un crucero muy satisfactorio, con muchos resultados. Y posterior a este ya comenzará la segunda etapa de este crucero, donde hay cambio de tripulación. Algunos científicos continúan, se mantienen algunos objetivos, lo que es el estudio de los cañones, y será muy importante también poder acceder a la zona de la Triple Unión en la Patagonia chilena, en la zona de Cabo Rapper frente a la península de Taitao, donde se juntan tres placas tectónicas, un lugar muy interesante desde el punto de vista geológico y biológico, y donde existe evidencia de la presencia de una chimenea hidrotermal, por lo cual será muy interesante poder documentar, filmar esta chimenea hidrotermal en directo, así que esperamos poder verla.
Perfect. Yeah, go for it. Perfect. Awesome. Great job. Nice one. Placement. That's it. Shop's closed. We're full.
Oh, yeah, of course. Sure.
Grove Street bus is full. We got it. All right. Nicely done, Ben.
Oh.
Fortunate. Now what?
What a good question.
We're all full of rocks.
Yeah, we've got the
so we don't
rocks
have
that
to
we
do any
need.
more
We've got the
rocks.
push cores we need.
Yep.
It sounds like
Nobody has explored up here yet.
let's go up there. I think.
We had we started to explore, and then they wanted to not fill this, and we didn't really.
Okay.
There's a little
Yeah, because that was
cut
above
through
the
there.
seafloor, right?
That's
We can
right.
actually go. So maybe we can go to that station.
You'll be the first one to see.
Station.
New ground. And towards the north end.
The mapping team's going to take over at 6.
Great.
Yep, go for it.
Bridge updated position.
Very north.
Amanda, do you want to do an introduction?
Sure.
Of yourself.
Yeah, sure. Hi there, I'm Amanda. I'm substituting for Elohio, our chief scientist, as the dive lead right now. We are currently exploring a 2,700 meter methane seep site off the coast of Mocha Island in Chile. We're finding lots of promising biology, some really interesting mounds that we think might hide some hydrates underneath them, methane hydrates. And we're here exploring these for the first time.
Amanda, where are you from and what's your background?
Yeah, I am from the great state of Minnesota. I'm a microbiologist, so I've been examining the microbes that live in the sediments here, the ones that eat up all the methane so that it doesn't reach the atmosphere and cause a bunch of global warming. And we're finding some really interesting stuff. Is the plan to still come up at 8? Or is that
7:30,
been
I
shifted?
think we're leaving bomb.
We're
We
leaving
have to
bomb
be
at-
on deck at 10.
Okay, on deck at 10.
Yeah.
Okay, great.
That's my understanding.
All right.
We're gonna see how fast you can pull samples off the ROV and turn them around. It's a mad race.
Yeah, wow, it's gonna be a mess.
Yeah.
So it looks like we're on the southern edge of one of these mounds right now. I think this is roughly where we came back to the seabed last time.
Amanda, have the inset in there.
Oh, great.
We don't have the latest data processed in there yet.
The M3.
Yeah.
Yeah, the
Adam needs to
hour
step
long.
up.
That's right.
Let's just blame Adam.
Adam on like a 4-hour nap. You gotta step it up or unlock his computer.
Alright, so this is kinda like new stuff here.
Yes, I think.
Kinda see some, it looks like there's some matting down
Yeah.
here in this little hole valley.
Yeah, there's a depression that should be.
Oh.
Yeah, right there.
This looks like
Oh, maybe that's trash.
definitely trash. Is that a newspaper?
I don't know, maybe.
Oh, there's a newspaper.
Oh my gosh, it's a newspaper.
Can we?
Yeah.
Can we pick it up?
That's a garbage bag.
Oh my goodness.
That's gonna break apart. You can look at it.
We got a newspaper. This is gonna be the new
You wanna read it?
thing that goes
Yeah, let's
Can someone type it?
Yeah, I'm gonna see if I can look up that headline.
Yeah, look up the headline and see if we can find a date on it.
Los sueños.
42 kilometers.
Chilenos, el marathon.
Olympic.
Olympics. Oh, it's about the Olympics.
Translation? Translation? Teacher?
20. It has Rio 2016 on it.
Does it?
Yeah.
So it must be later than that.
Olympic. They're losing?
Can
we turn off the laser, please? Thank you.
We can read articles.
That lady is underwater famous.
Yeah.
What is that?
You need a computer.
No sale, ¿no?
I think it's in advance of the Rio.
It's the future.
No, because it says Rio 2016.
Where? When I see that.
Rio 2016, but they might be talking about it. But they have a picture from Beijing, 2008. Rio.
2000.
Wow.
It's been here since 2008, is what I say.
This picture, I guess, is from
Yeah, the
2008.
picture's from Beijing Olympics.
Okay, yeah.
Could we just give it a little?
Yeah. Suction have an inverse. Wow.
Subsea times.
Just got that.
The slow chuckle around the room.
Acrax, clams.
Oh man, look at those rocks. Cool little spot.
Is that a mat on the left side?
Looks like it. Let's see if we can. There's a bunch of it along the side.
Yeah.
And the bottom too.
Here on the bottom,
It's
too.
just gonna kind of land out here and we can have a look, a little zoom around.
I've seen these questions before. I think the volcanic eruptions.
Is that right?
A couple of years ago, small ones.
Oh, yeah.
That sounds official then. I like it.
More of the skeleton shrimp, too.
There's shells, teacher. There's more shells.
We've still got a suction sampler
Yeah.
left.
No?
You want to? Great.
Okay, Ben, picking the starboard rail out. I'll
In the
have to
same
move
one?
for you to get
Same
that.
one?
Same one, SS1?
You won't be able to reach that, but just grab the hose and then I'll put it.
Maritza, do you know if these snails have any of the sulfide oxidizing symbionts or why they live in these environments?
I don't know what is the
I think we're going there, I don't think you can reach it, so I'm just gonna head over. Because it's also down. Right here now, I'm just gonna set down. What jar are we wanting to see? One. One? All right, Ben. I think that's them right there, right? See 'em? How's that? Do what? Grab out for stuff.
You wanna go? I'll go get a food. Yeah, of course, I'll give you the chip. Should we do something?
I just
Okay. And we need to close. What
We need to close this hands before
here. So before so.
Yeah, so,
Okay.
and I'll be back out.
Yeah, don't worry, you wanna take your time. Oh, snails.
Ready? Okay. You're gonna get really far away, Ben. There. Okay. Just haul it back in. I'll bring you close. I'm sorry. Right there. Is that the rock you're moving around?
We want to try again?
Yeah. I'm okay, you can see him now. You ready? Yeah, it's on.
Which number is?
One. There's two of them. Just down below.
So what do you? One.
That's where there's other ones like this, yeah.
Should we two?
There you go. There you go.
setting or anything?
Let's go.
Down there, up along the side.
Yeah.
See it fly around that way.
Good idea.
We've seen areas like this a little bit young, still fresh.
Oh, yeah.
This one's really old.
So we have 8 and 2 available. Okay. What's that? It's a big
What?
fish.
Where?
It's a fish.
Oh, it's a fish, yeah.
Yeah, it's a fish. This is over where we mapped earlier.
Yeah, he doesn't say. Yeah, we're heading that direction.
Yeah. It's a lot of depressions.
Yeah.
Bridge, I moved you north a little bit, 20 meters or so.
20 north.
These holes are. See these holes? Some sort of trap. There's a rock in there.
Ah, there is a rock.
Yes. Yeah, that was Lisa's last rock.
Había un día con el mercurio, no es tan viejo, es de la
What's
boda.
that, buddy?
Que está en el fondo,
Where?
no se había degradado.
To starboard, yeah. Yeah, that's the way I turn it.
Luego soñó. You're part of the newspaper. Yeah.
Porque necesito saber cuándo se van las botellas.
You make it close up here.
Yeah.
Can we collect?
With what?
Correct quivers? It's not, oh, it's too big.
Probably too big for a quiver.
¿Qué te dijeron?
Puedes poner aquí con las clams.
No, no, correct quiver.
Pero ha dicho que es muy grande.
No, it's too big for correct quivers. No,
No, okay.
it's too big.
I don't know what you meant.
What do you think?
But hey, just a second. Can you put it here in maybe 1A? Because it's an unibrow.
Yep.
No?
Maybe rocks. Yeah, put them in the rocks. No, that's what Lisa's samples for. You have one that arm and scrape the nose. There we go.
It's 15 centimeters.
Okay.
Yeah. Big boy.
Yeah.
Basketball.
Oh, it's a unibrow. Super nice.
Top that, should that.
Oh, it's a scooch. It's huge. All right, continue?
Yep.
Okay.
Ah, it's a sea flag.
That's one of those in a while. Starfish. I suppose. I don't know where else to go. I'll try and turn back down that valley over there. See what's in there. Were they doing any more mapping tonight?
Yeah, the plans to map what's
When they planning to map at 6 o'clock, because I tried to ask them, but nobody's here.
I thought I heard 6. I read you over here.
Yeah.
And then off bottom it's 7:30, I believe.
Okay, so before starting mapping, we need to close the past 6 o'clock.
Okay.
Because we need to be very close to the. We have.
Is that on bottom?
Yeah, like now that we are very close, we decided, we find something.
This direction.
Yeah.
So then you'll just have to,
The previous one was nice.
and you'll just have to pass that through. Did you catch all that though?
Nope. What do we need to do?
More stuff to do. So you were saying you want to do Niskins here at this site. Well, do Niskin.
Yeah, the last two, because I completed this one as well. If we start now the mapping, this is the site we'll be together. Now, because I asked you before I went here, but you couldn't say because otherwise I could close before with the clams. It doesn't matter, we can close here, there are other organisms, so we close it, 2. Yeah, and then we start the mapping. Thank you.
And Dr. was just saying before the mapping starts to do Niskins. You're okay with that, Howard?
Do you want to stop doing here?
You want to finish it now?
If we start to mapping now, yes, it's better.
Okay, what's Niskin now, please?
Carbonate's skeleton, let's call it in this way, okay? Because it's carbonate is able to record the carbon derived from the methane and the isotopic signal of this methane, which is very characteristic with respect to whatever else you have around. So, if they are alive, which is difficult to be, but if they are alive and are able to live a little longer when you have methane escape, which is an extreme environment for them at least, they are able to incorporate the signal of the methane during their living phase. Then you can date that foraminifera with a specific age, and that's the way that you have to say, okay, there was less or more methane at that specific time in history. But most of the time this is not happening because difficult for them to live there is a very ephemeral thing, so you need to use a very specific technique to look into that. What's happening is that all those big carbonate crusts that we keep seeing on the seafloor, they are actually growing as well around the skeleton of those foraminifera. It's called carbonate overgrowth, and that happens after they die. Most often that's the things that you have and the things that you can measure relatively easily when you look at the foraminifera, but they don't give you the age when they form. So the foraminifera itself will have an age, maybe 1,000 years, but the carbonate around it maybe is done yesterday or in a much more recent time. So there is this time lag between those, and that brings a layer of difficulty in really dating when the methane was escaping or not. But in the easiest way, you can say when everything started, if you are able to find a spot where you don't have any more going down deep in the sediment, you don't have any more signal linked to the methane in the forams or in whatever other parameters you're looking at the specific moment, you said, okay, that specific sediment deposited before there was any kind of methane emission, okay? And that's it's gonna be it. At least you give a minimum age to your seepage system, and the maximum, it can be done at the maximum level as well. We have often seepage systems that are not active anymore, and if you are able to see what's the last sediment age that's been actually affected by methane escape, you can give a time range at least in the past for that. That's a short version of.
That's the short answer.
Yeah, the short answer.
It's
Then you're now adding sedimentation rate and boron availability
Yes.
into the equation for the, you need those to factor in for the age. Well, and I guess now radiocarbon uncertainties. Is that
Correct. It's most of the dating things or techniques that we use to date sediments or a stratigraphy of a place are based on carbon. And carbon is one of the two things that you have in methane. And the signal is very different. So you need to apply a lot of corrections and a lot of just taking it into consideration that the signal that you see when you try to date maybe has been altered by the methane presence itself. That's why it's very important to take an equivalent length of sediment from an area that you are reasonably sure has not been affected by methane. Because at that point you can date everything inside of that very nicely, and you just do a geological correlation between the two different sedimentary columns. And so that simplifies the way of dating the sedimentary columns. Because also the other way that we use when we have longer periods of time that we want to date
End of
is
line.
oxygen isotopes.
End of line.
But if you have gas hydrates, the water in the gas hydrates, when they dissociate, tends to release oxygen that is heavier in the isotope, so it skews the signal as well, and so it's much less easy to calibrate with an age the sediment that have been affected by that process. So
here that might be different. So here specifically that might be different.
Yes, that's why we really need to see what we can get from what we have and try to make the best out of it. Sometimes it's impossible. Sometimes you just give an estimate of things. And of course, you need always to remember not to look just maybe at a single kind of data or a core or whatever it is, but you need to look at broader to tell what's the geological history of this place or all these kind of things, and then putting things together. So it's not easy.
I know that some efforts to use radiogenic isotopes, uranium and thorium decay, to age date carbonates at Seeps in other parts of the world have placed them into the order of thousands to tens of thousands of years old, so maybe back to 15,000 years before present. But those were relatively shallow compared to this site, and they were likely locations where hydrate was disassociating the seafloor, but they weren't necessarily controlled by geologic features like faulting. In this site, we're much deeper, and we likely have a fault running underneath the site where we are right here, which could suggest that the site may be substantially older than that. Those features certainly are much, much older. And so this potentially could be an older site, but as Davide said, that would require looking at the sediment, maybe looking at the carbonate rock, a wide range of methods to sort of try and generally constrain an age date.
Yeah, indeed. And that's when you need to consider the geology where you are in the equation to try to define things.
Yep.
Because the sites here are deeper, there are the faults like Adam said, but they are profoundly different from the seepage sites that we were before.
Yeah.
Completely different kind of geological system, and they don't work the same way. So you really need to evaluate on a case by case how you do things.
Davide, I can't remember, do you have any uranium thorium plans?
It depends on carbonate availability and things. That's definitely something that can be done. Of course, a sample
End of line.
to be taken with that in mind.
Yeah.
We cannot just potentially just do that pieces that we found on the seafloor, and to do that is to take a sample that is as thick as is feasible and break it off when it's still in place and note which one is up and which one is down. Right.
So really counterintuitive to what we think of as geologists a lot is we tend to think that the deeper something is, the older it is.
But in carbonate rocks, it turns out that the deeper you get from the surface in the rock, the younger it gets. So the rock is actually, you can think of it as growing downwards into the seafloor sediment. And in fact, a lot of age dating has shown that the age is very significantly throughout the carbonate rock platform. So it's a very complex to try and get at rates. And
if you would like it, Adam, I have a
ages.
database of every uranium thorium measurement on cold seep carbonates.
Oh, I would love that.
Yeah, I have all UTH data that I've been able to find and pull.
Wow. If you're looking for a Christmas gift for
there you go. That was an easy buy.
We are doing something in the Gulf of Mexico. We are with some colleagues from China. We are collecting all the data available, plus creating new one carbonates around all the seepage of the Gulf of Mexico. And something is interesting. We put up on the abstract a couple years ago, I think. So, yeah.
I remember seeing that. Actually, I think that was right after.
It was the
Do you separate those?
remote kind of thing. I don't
That
know
just
if
maybe
that's
20
close to
some,
22.
12 or 124.
Yeah, yeah, because it was, I think it was after the one where I met Gracie.
Yep.
Yeah,
Start a line.
Yeah, probably.
Yeah, because I remember seeing that.
There will be some
Yeah, no,
areas
we've been
where
trying to
mix
figure
it
out. We should talk more because I'm Waifu and I have been trying to figure out a good paleo section, and it's the good paleo section is South China Sea.
Yeah.
And
yeah,
and we want to apply the Cat 47 proxy that I'll be publishing soon at some point to that, because that'll put other sorts of numbers on carbonates, which will be really nice.
Yeah.
And another good way to date seepage activity in cores is if you find small pieces of carbonates layered through the maintenance.
Yeah.
So you can actually date the carbonates and do that.
Yeah.
It's a very nice science advance paper. I think it was done by Germans. I think it was at 200 meters long. Core drilled with that lander system.
Yeah.
So it's different level.
Yeah, no, exactly. Well, the South China Sea ones are the 60 meter.
Yeah.
Those cores that I've been looking at. Yeah, and those are 130,000 year.
Yeah.
Yeah. Last interglacial. Yeah. Those are cool samples. What happens when you put a bunch of geologists in a room and we look at water while we're looking at seas? Yeah, I have funding for major and minor elements and isotopes, but not radiogenic for my samples.
Yeah, and that's where the money starts to ramp up.
Mm-hmm.
Yeah.
Yep.
Yeah. That could be done. Though.
Yeah. Yeah, we'll see. When I'm taking isotope samples, I'll make sure I keep good powders. Yeah.
The important thing is that you take for uranium thorium is that you get some background sediments because you need to correct.
Thorium correction.
Yeah.
Yeah. Yeah. Well, I know a couple of people who've taken background cores in this area.
Yeah, that's the worst, right?
Yeah. What are we up to now? 20 push cores a dive.
Listen.
And hey, background actually doesn't necessarily mean the same as background for everyone else when we're talking about.
I think their background is like 2 meters apart. My background is 2 kilometers apart.
Yeah, no, exactly. Their background is we don't see clams. Okay, we're good. Only if you find the clams first. This is where Sage comes in. Except we've been sitting on top of clam beds and haven't seen nothing.
Because the organisms are doing the hard work.
Yeah.
It's pretty excited about the free tree cut yesterday. I thought when I saw the picture of the one on the way doing that, we lost it. And I know that the shadows is gone. They pulled it out of the fridge and turned it into a tree just to show me. So excited about it. So it was straight to. It's crazy stuff.
Davide, can you talk a little bit about, in our wonderful mapping podcast,
Yeah.
the goals of a long core at a site like this? Why is it important to try and get a core here?
Okay. Well, there are multiple goals if you want on having long cores. It really depends what your goal is. But one of them could be to really pinpoint this transition zone where the methane is all consumed. And because that gives you an estimate of the rate of the methane flux. Of course, if we core straight on top of where we have the hydrates, besides not really being able to recover anything, but if we would
0
do that,
centimeters.
I would expect what's called the sulfate methane transition zone, very, very shallow, maybe even at the seafloor in extreme cases. But this is sort of the hotspot of the spring, the seepage, which is a very small location. The seepage system is actually much broader and not always includes fauna on top of it, or visible bubbling, or whatever you are used to see now. So, doing a push core with ROV gives you a good image, very high resolution of the first 20 centimeters. But there are seepage systems quite widely studied, quite active, totally analogous to what we are looking at today, with hydrates, with active emission, etc., where this transition zone is right few meters next to the most active area, 6.5 meters below seafloor. And that's the only way that you have to measure that, is to long coring. If you move another kilometers or even few hundred meters, that's going to be 12 meters. Okay? So that really gives you an idea of what's the distribution, in case you have multiple cores, of this zone, so of where the methane is coming up more or less faster or slower. That gives you a very good thing. Besides trying to get the oldest record on methane escape as you can, because as we said, the deeper you go, the older you get in terms of sediments. So ideally, you want to really hit the moment when the methane escape started. And if you're lucky enough, you do that. And that helps you to really understand how the system evolved from that moment to present. And why we want to do that? Because we still have big questions or big unknown things on how, from a geological point of view, this system works. And it's nice to know all the ecology associated with that, but on a broader scale, we need to know why they are there, the seepage systems, and how they work. Because at the very backbone of everything, we have methane seepage because some sort of geological thing caused them to be there, or caused them to be existing at all. And by trying to do this kind of reconstruction, we can understand better what we can expect in the future. There's been a lot of discussion about, methane is a very potent greenhouse gas and that can be have an influence on climate. That's a very sensitive topic, and the role of deep sea seepage in climate is something that is almost certainly minor at this stage, meaning that all the methane that is escaping from the seafloor is very rapidly consumed by whatever process into water column. This means that only a very minor fraction that we have question marks around it, how much it is able to reach the water surface and so go to the atmosphere. So that's one aspect. To have really important input of methane into the atmosphere, what we need to have is a shallow site, maybe 100 meters or even less, with very large amount of methane escaping at the same time. And that has been observed and hypothesized in the past for big global scale climatic events that in a way or another could have contributed to that.
Shout out PETM.
Yes.
Yes. So, sort of minefield.
Yeah.
This
Yeah.
kind of
Yep.
this. But definitely the things that we can say is that reconstructing the evolution and how much methane is emitted on the seafloor is extremely important to really understand what is the chemical, physical, and biological aspects related to the water column in that area, because all the microbially mediated processes that act on the methane and in this interface that we set create a lot of byproducts, a lot of sulfates, and a lot of other things that eventually alter the chemistry of the bottom waters, and in some situation can create changes in the pH. So in alkalinity, or acidity in some way, they can fluctuate that, and so they influence what kind of organism that can live there or not, plus having a lot of other things. And there is, besides this more knowledge and sciencey kind of aspects to that, the study of methane seepage or seepage in general and the revolution is extremely important from an industrial point of view, because they are, if you want, an hazard for any kind of anthropic activity that we can do in here. A lot of seepage can create blowouts, totally natural, and they, in simple word, loosen up a little bit the shallow layer of sediment, so they are less structurally sound if you want to build up something on it. So we really need to be very attentive of these things, the evolution, and why they're there or where they potentially form when we want to put any kind of things in this area, from oil and gas platform or pipeline to offshore wind farm to really anything that requires some minimum contact with the seafloor. That's it. And as I said before, the energetic aspect is also something that is important. This is one of the first things that you look at when you look at new frontier basins if you want to explore for oil and gas resources, because it's the easy way to say, okay, there is an active petroleum system there, let's look more into the detail. So these things are used across the spectrum, from science, climate science, ecology and biology, geology, and industry applications.
It is worth noting from an industrial standpoint, there are locations where methane seeps with bubbles coming out, like we've seen here, have been used as an energy resource. Those bubbles have been captured and put directly into a natural gas system and used for things like heating homes and cooking. So these are potentially an energy resource, although in most cases they're not economically feasible for that purpose, but they have been demonstrated as such. And then, as Davide mentioned, there's potential for geohazards. Overpressurization of the seafloor can result.
Front lid right there, the next to the margin, we'll get this line and then we'll just come up. It's like going to be...
We need to be, I think, on deck by 10.
Yeah.
So if we need to bail on part of this last line.
It's 20 past 7, so we'll be off bottom like 8.
Okay. So end of line.
End of line. This might be the last mapping, Adam.
It might be.
What's our arm? I know the bottom has to be.
Okay, and we're moving.
It's a negative wall upside down.
So Scott, if this is the end of Sage's time on this cruise, what's next for Sage?
Oh, it's not.
Not,
No,
well,
it's
this
staying
leg
for
of the
leg
cruise, I
2.
should say, yeah.
That's why it's not on this dive.
So, exactly.
We're going to the triple junction.
So, a completely different sort of environment.
Completely different sort of environment. I'm actually really interested for what the methane is going to be, but also what we're going to be able to total gas concentrations like oxygen concentration and total gas concentrations like oxygen concentration and temperatures affect our signal response has been a really unplanned but convenient part of this cruise for me. And the triple junction offers another deeper and in other ways distinct sight from these ones that we've been on. So I'm intrigued to see how it handles, especially our smaller unit. Beyond that, I don't know. There's someone sitting 2 seats to my right who's looking at me. She would really like to have it on a cruise that I believe is next May.
Yeah. I'll be there too.
Are you gonna be there too? Maybe
we'll
I'll have
both
to
be out for that cruise.
I'll find out from Anna if she'll put me out there. Yeah. No, I mentioned that to Anna.
Yeah.
Yeah. I don't know if Mandy's talked with her. But yeah. There's time.
So I think we're going to put Sage on an AUV and.
Yeah.
That would be awesome.
I think did you ever put Sage on the center?
Yeah. Sage's been on
So
Sentry.
it fits.
Yep. No, it fits. I'm 90% sure Sage has been on Sentry. Not with me on the deployment, but yeah. I never really know what our field schedule is going to be before like the start of the next year. Like I'm sure Anna has some idea of if we're doing anything early in the year, but
yeah,
I don't know what that is.
I keep forgetting to ask Mandy.
Yeah.
But definitely.
Not start a line.
Just get some extra covenant in this corner.
That's right.
What's a triple junction?
Ooh.
3 tectonic
question.
plates
Yeah.
or something?
Yeah.
What do the Azores have? Don't they have something special like that up there? Is there some tectonic business up there?
Not the Azores.
There's a lot
They're
of
a
work.
hot spot, right?
Yeah. The Azores don't have a triple junction. They're
No, they have
no.
okay.
They're hotspots.
So what's a triple junction is 3 tectonic plates?
3 plates coming together.
That's the deal.
Yeah.
Yeah.
There are not a lot of those.
What happens there?
Stay on board and find out.
Yeah.
Stay tuned.
So you got 3 plates. Yeah. Splitting apart.
You'll have some live streams.
Yeah.
Tune in.
Dive streams.
Dive streams. Levi will be sitting at home bringing it up, doing welcome back to dive stream. Oh, look at that drift.
Oh my goodness.
Oh.
Tokyo drifting.
Oh. Love it.
Start a line.
Start a line. Start a line.
Wow.
Everyone hang on.
Punch it. It's awesome.
Levi, if you zoom in close enough, those look real fast. So, but yeah, Sage's. We have 1 unit on here, but we've been working on scaling up. We currently have 3 functional units. So it's starting to be the case that we're getting on multiple.
Yeah.
I don't think we've done a multiple cruises at the same time yet, but we've multiple stages on a deployment. So Mary just had them out on Hercules and American Samoa. Literally, they got back 4 days before I left for here. So that was tight.
But technically, our cruise is still not officially scheduled, which makes me a little anxious because it's like 6 months. But yeah.
Yeah. No, I don't know.
In theory, we should be doing that.
Yeah.
It's the very last Endeavor cruise ever.
Oh, we should definitely.
Yeah.
Shut
it down.
We need to go party.
I can just drive it over to Rhode Island and stick it on there like in an hour and a half drive.
It's easy compared to
flying to Santiago. Yeah.
We need to do a bathing ban because
Yeah.
there's impressively low amount of baths on that. 'Cause,
Yeah.
but yeah,
Plan is
that-
to be on deck about 4:10.
Yeah. The what was I gonna say?
What's that?
Yeah, I don't know what our field schedule is. We've taken
We'll see, we may-
for the last 2 years. We've had one on Kronprinz Haakon with Juliana
We may
Panieri
be
out
able
of
to
Tromsø.
this line about half.
I took it out. Beckett and I took it this May. I don't know if they're even planning on doing a Haakon cruise on the mud volcanoes up there next year. If they are, I know Juliana's probably going to want Sage on there because it was great this past time. But we'll see. I mean, everyone likes it, everyone wants it, and there's a reason we're ramping up production of it.
They have their own little microplate.
Yeah, look at that. Look at that triple junction. Oh, for the Azores. What?
Africa and yeah.
Azores microplate.
Is that caused by the
I don't know, I'm not a
hotspot?
geologist. But I just was there, they were excited about it.
Levi's gonna take over the Falcor 2 and drive it to the Azores
It's
to
all
go
right. That's
dive
what
on the
I
triple
said.
junction.
You guys are going around the horn anyway, right?
Bridge, new waypoint?
Yeah. Limit of the grid. Wait, that's big.
Could be the world.
That is the world. Wait, Levi, how far away are those other markers?
Look at the coordinates.
Yeah.
Coordinates. I leave you alone for a minute.
Okay, what is going on?
Don't worry, I don't want to know what we're trying to do now.
We're trying to maybe go to Highly recommend.
Yeah.
Might take a while, so that's
I have to calculate the transit time on that one.
Yeah, Adam, you've been doing a lot of transit time.
Transit time on that one.
Yeah, Adam, you've been doing a lot of transit time calculations on this dive. Can you,
Can we be there by next summer?
But do we want to do 2 hertz or 6?
Definitely 6.
We're mapping the whole way.
Can we do a bongo net too? Halfway along.
Also do a bongo net.
But only one at 2 knots.
Dude, we could load up on fuel, load up on food, and do the world's longest ROV dive. And M3 mapping the whole way.
Yeah,
We're
the
gonna
whole
do
way.
our NASA's too. We get shipping containers full of food, no problem. Diesel, no problem.
Yeah, let's load the aft deck with containers of diesel.
Helicopters can come and bring us supplies. Well, we keep going half of that.
Be well worth it.
By the time we get there, it's gonna be a whole other group of people.
Yeah.
Well, it's like the Enterprise.
Yeah. I was gonna say it's like any of the like long-term space travel novels where it's like 3 generations later they get to Mars.
Well, that should stay down until we lose a cop. Pretty much, yeah. So we'll have to put big zincs on, because zincs are gonna
Yep.
go. Ground fault system.
Yeah, those. Oh, come on, Levi. Sage isn't on there. Don't worry.
Ground fault system.
All right, Davide, question for you. We have an option here. We could curtail this line here about deck to be up a little early to preserve maybe a half hour for corn tonight.
Okay.
Or we could finish out this line.
That's a tough question.
That's a tough one. That's two competing priorities.
170 meters and you're going 20 minutes.
But you have like 70 meters,
Yeah, this beautiful
24
square.
meters per minute.
I know it's not going to be a beautiful rectangle.
I got a surveyor to do that.
I know, trust me, I understand, but
I don't know. Are we looking at anything special here with the structure? Or if the structure, how
Just
is
did a
it?
line.
Look how special that looks.
yeah.
It's, it's not even me.
I'm just giving you options. It's like the menu.
Those are the turns.
And
then the
I'm talking about 9 minutes.
9 minutes to the end?
Yeah.
Oh,
Yes.
never mind. Question redacted.
What was?
9
4
minutes.
meters per minute at 0.4.
9 minutes to finish.
Kinda, so I should be.
We're gonna finish.
Yeah. If you want to make up time, let's just haul on the winch faster.
60 meters a minute.
Wasn't Dan saying something about the bridge?
So we're supposed to suction something over there?
Oh, we are.
I don't know. Well, they gave me two pictures and they look the same.
Yeah, we've been looking for them already. I think we know
Polychaetes?
what we're looking for.
Oh, Mike's gonna find a pelagic polychaete.
We found one yesterday, but it wasn't
Oh,
the
yeah.
right one.
Ah,
What happens if we find one in this last section of mapping? What do we do?
Dude, we bail the mapping line.
Yeah.
For a pelagic polychaete?
Dude, look at what the mapping lines we did. We're good.
We can.
We bail.
We bailed for.
Depends on how cool it is.
We bailed for that onyx squid.
Yeah.
We just totally gave up on mapping, and it was worth it.
Yeah, that was cool.
I think you gauge the room, right?
Whoa! It's like we can probably walk away.
For that polychaete.
This is the wrong room.
For
to
polychaetes.
gauge for pelagic polychaete.
we're not gonna give polychaetes much
Three
love.
geologists and a microbiologist who used to be a geologist.
It comes out like, oh, that's cute.
okay, bye.
No, that onyx squid with the eggs was once in a career film.
Oh yeah, no, that was unquestionable. Like it wasn't even a question.
And
Just
then we saw
go.
a second.
Like, you guys aren't super excited
Yeah,
about
Yeah,
it's a-
we had two.
Like,
When was the next
The
one?
second one
A
didn't
few
have
days
eggs.
after, I think.
Oh,
Yeah.
boring. Just kidding.
No, it had eggs. Yeah, it was, most of them had a hashed out, but near the tentacles it still had eggs in that.
is that the one that we mulched?
I was worried about that. I don't think we did. Do you think we did?
I don't know. I was
Yeah,
thrusting
I don't-
up and it went above me.
I was unsure.
I don't think it was that one.
No.
Yeah,
It's
no,
not.
definitely not.
They're faster than your ROV.
That's true.
Yeah,
They probably could
but
shoot me right next to it, which would be, yeah.
we did collect that egg case in the slurp.
Is there any eggs in it?
I don't know. That would be a question for Greg. There were. I mean, we could visually see that there were still eggs on the upper part of the case. So I don't know. I don't know if it was a portion that we slurped or if it was the whole case. But well, that's like how Mike keeps finding once in a career flying spaghetti
Yeah,
monsters
he's
every
the-
fifth night.
Water specialists here. Like 3 of them already.
Should have slurped it.
Yeah.
I don't know, the way it was stinging that fish. I wouldn't fancy handling that fish.
Yeah. No, thank you.
Nope.
Open bio box and just get it in there.
Yeah, just come up under it with an open bio box.
Is there a swimmer? Yeah, there's a swimmer.
It's been done.
I saw it in there.
Yeah. When you're trying to catch like really little gelatinous things in the bio box, and every time you approach it, it like drifts off to the side.
I've wanted to make the bio box have a
No, we still
the drain tube at the bottom, and then there's a vacuum you can turn on. So
It
like,
sucks all into the
so,
box.
yeah, it's just sucks stuff into the box. Like it's nice, slow, gradual suction. You know what I mean? Stuff wants to go in there rather than like not want to go in there.
So
We're not gonna be done. We're just gonna have to walk around.
Yeah. Should have a little power on the section.
Oh, man.
We're about to start coming up.
Yeah.
this line.
We didn't see any bubble line. We didn't see any flares.
No, we didn't see
No.
You're good. Too busy, yeah.
We also haven't been paying that much attention.
I've been watching.
Oh, okay, I've been paying
That's
attention.
why Adam is the front row.
Yeah, that's why he's dive lead.
And then do a bit at the start.
Or the peanut gallery in the back.
Tell me your base. It's
Tell
a
you.
Teach me everything.
Okay, so, can we wait until we hit end of line? It will be our last mapping line. End of line, end of survey. That'll be about 3 minutes.
Oh, I'm leaving 4 to 7. What? I'm leaving 4 to 7. Yeah. According to the garden, so because it's sampling, so we want it, and you're going to the black lights that we won't sample, so like, it makes sense of what it is you're paying. No factors. What the rest of what you were gonna say?
I just, nothing you can say.
Oh, okay.
This is stage dedicated.
It's just a little chill.
Yeah.
You said it really cold down there.
Yeah.
It's a highly accurate.
It's minus 82.
Yeah.
Sure about that? You sure about that?
Yeah, the CTD is actually off by 84 degrees. The temperature probe is right, everything else is wrong.
It's the IPS measure. IPS.
They're going into the house. Oh, if I weren't at Seep doing Seep stuff, I'd be leaving and stuff the next 3 days. So I go in and be late.
As soon as we're coming up, we'll go over the computer. It's quick, it'll be short. Keep running copy files.
How mad would people be today if, as a joke for the day after Halloween, I put on Christmas music?
I will throw you off this phone.
Yes. No.
And if we tell Phil, I think he'll be okay with that. All we have to do is make sure Levi puts the new waypoint in the opposite direction, and you'll be left behind.
Put it in the Azores.
In the Azores.
So far behind. I've always wanted to learn Portuguese.
No, just stay here. That's where we're going.
Bring me back some pastéis de natas. Obrigada.
All right, end of line. End of our mapping survey.
End of
We
survey.
can haul away.
Right.
You guys go.
We come.
I'm gonna play that graduation song.
What's that? Yep. All right. So this is the end of our dive for today. We've just wrapped up our M3 mapping in preparation for a dive, which I think will be starting in roughly 4 to 5 hours. tomorrow morning. And so we'll be back at this site for another dive tomorrow morning, and look forward to seeing everyone online back to join us on the Falcor 2 for more exploration tomorrow.
Goodbye. It's just goodbye. Good. It took a little while to make it through my brain. I heard a bye.
Update on watch times?
Nope. Except there is one update. Aaron is leaving 47.
Your ship
I
is so
think
big.
Marisa is on the 4 to 7.
You're isolated from all of the
And then I think it's
noise
Lisa, and I think
information.
it's Greg. I think.
Okay, so we're just shifting.
I'm used to like
Update is no one sleeps until
Yo,
No
Sage
one
isn't on this dive.
staying.
Sage isn't on this dive. We get to sleep.
The irony is that now we're up in the shallow Mocha. We're gonna have so much less to do.